pharma research. Means what? The in analysis also, in formulation development also. So in every domain, you quite be needing these computational tools. Nowadays, you often find any formulation development topic with some binding analysis, or you can say molecular docking analysis. So that's in this additional data will increase the acceptability also, and will improve some or will definitely improve the publication quality or research quality also. Now, before start with this computational tool, I must discuss this chemoinformatic terminology with you, because all these computational tools are run around these chemoinformatical tools. Now, what happens in this chemoinformatical tool, or what is the basic fundamentals of this chemoinformatics? This chemoinformatics is nothing but the core gathering and the systematic use of chemical information. Nowadays, this chemical information in terms of computation language is often calculated or transferred, and this chemical information and this application of this information is utilized to predict the behavior of the unknown compounds in silico. That is the fundamental application of chemoinformatics. Nowadays, this when it all this chemoinformatics journey has started, these computational tools are strictly restricted towards the medicinal chemistry part, or you can say that some drug discovery part. But after continuous upgradation of computational tools or language or databases, these computational tools are nowadays commonly applied in phytochemical research, analytical research. Nowadays, you can correlate analytical behavior of any substance with the computational tool also. That is also known as the quantitative structure activity quantitative structure retention relationship, where you can develop a computational models to predict the retention behavior of the drugs or unknown compounds, or you can utilize this information for the mobile phase optimization or mobile phase selection, so that the experimental work, or you can say manual work, can be reduced, or the uh, wastage of the chemicals, or overall the cost effectiveness can be achieved in case of the analytical purpose. In case of the formulation purpose, there is nowadays you can say QSPR technology has been continuously evolving, because that is the quantitative structure property relationship, in which the properties of the polymeric matrices, you can say, or drug or the excipients have been correlated with the release profile, or based on the mathematical models which are generated, the you can say the formulation part, or the formulation component can be easily carried out. So it leads to the further optimization, or it can this can also minimize the um, overall human efforts, or the cost, or it is method becomes a cost effective one. So now that's why this chemoinformatical purpose or chemoinformatical tools are commonly used for this in silico modeling, or in silico technology, in silico understanding of the chemical behaviors. Normally, when you see this chemoinformatical terminology, is often correlated with the data and the analysis of that data with the predictive analysis, analyzing that data for the prediction purpose. So that is a basic utility of this chemoinformatic purpose. As a hardcore medicinal chemist, before discussion of this all the chemoinformatic tools, I must discuss the what is the exact or medicinal chemist thinks about the drug candidate, because the drug candidate often we call as a structure or all those things. But normally, when you see a drug candidate, it is normally a drug is nothing but a ligand molecule which binds with the biological target, and this target is often a protein, enzyme, or receptor. And by binding with that protein, enzyme, or receptor, it either initiate a process or inhibit it. Means that ligand can have agonistic properties, also antagonistic properties. Normally, when you see this ligand and all the binding affinity of these things, the structure or conformation of this ligand is often important, and it should be a complementary to the protein surface, or you can say the active site, or size of that protein, or the active site, size of that active site, or you can say that this should be perfectly fit with the active site. And this often correlated, or this binding is often caused due to the favorable interaction between the ligand and the side chain of that amino acid with the active sites. And these interactions are often called as electrostatic interaction,
Excuse me,
hydrogen
sir.
bond, or
Sorry to
yes.
disturb you.
uh,
Your slides are not moving. You are not in a slideshow mode, sir.
okay, I'm in slideshow mode, but I think.
Actually,
Uh,
we are able to visualize only the first slide.
okay, okay. I'm extremely sorry for that. Due to some technical or I think there is this connectivity issue from my side, so it is visible now.
No, no. What you do,
Yes.
you
Next
actually
slide ला
shared,
जावा
I
ना.
think, a window.
Yeah.
You actually
Now it's
shared the whole screen. Then it is visible.
Yes, yes,
Uh,
yes.
Okay, okay. Sure. One second. Sure.
Next slide.
Is it visible, sir? Now?
Yes, sir. Visible आहे sir.
आता मूव होत आहेत?
नेक्स्ट करा सर. नाही मूव नाही होत आहे सर अजून इथून.
ओके. हा आमच्याकडचा जरा
नाही,
नेटचा
whole
इश्यू
screen
आहे.
share करा.
आता पुढची स्लाइड दिसते सर.
नाही नाही ते मी एक जस्ट मिनिट
Whole
सर.
screen share करा सर.
Shall I continue with this mode? It is readable from your side.
Yes, yes, yes.
Hello.
सर तुम्ही काय करा, तुमचं कम्प्युटर शेअर करा, म्हणजे पूर्ण ती स्क्रीन मूव्ह होतील, मोठ्या झालेल्या स्क्रीन पण.
बस बस.
तुम्ही शेअर जिथे करता ना तिथे पॉवर पॉईंट करू नका, होल स्क्रीन शेअर करा.
हाँ. आता है.
right one. Done.
मूव होते सर.
Yes, sir. Yes, sir. Now it is.
Okay.
We can see it.
Okay, okay. Sorry for the technical glitches that is occurred due to from
No
my
problem,
side.
sir. No problem, sir.
Okay.
Okay.
Okay. So that we have started with the discussion of informatics. I think uh, we'll continue with the drug candidate. So this binding cause with the this favorable interaction to ligand and the amino acid of the amino acid which are present in the side chain of the active sites. Normally these interactions are electrostatic interaction, hydrogen bonds and hydrophobic contacts. But why this interaction occurs? This interaction is nothing but the physiochemical interaction between the chemical you can say that the chemical behavior, it is a chemical behavior from protein side to the ligand side, or you can say the interactive atoms are there. So normally these are all the contributors of the disk structure of that particular system or the structure of that ligand molecule. So this structure often defines the binding capability, or the structure often defines the interactive behavior of the molecule. So we can predict the interactive behavior, we can predict biological activity with the correlation of the structure with their property, because this binding is solely contributed by the structure, or the binding is solely contribution of the structure, or the you can say the structural properties of that particular ligand. So if we are able to correlate, if we are able to calculate the structural property, so might be we are able to predict the overall behavior of the molecule. So this basic concept is commonly used for the drug discovery purpose in case of the structure-based drug design, or you can say ligand-based drug design. And this also have been utilized widely in QSPR, because in the QSPR, what happens, the polymeric property or the properties of the polymers is calculated with the help of some statistical tool or software, or you can say that the database, and these properties are correlated with the release profile, or the particular property that one has to be targeting. So we can easily identify which properties are contributing towards that which structural properties are contributor of the desired property. So we can modulate that property in the polymer matrix, or in the case under study, so that the or the result will be optimized in that case. So we can say the structure is the overall contributor of the all the physiochemical behavior of the system in the biological processes. So we can easily modulate the biological process with the modification in the structural properties, or in the chemical structures. Normally, in a fundamentals of drug discovery, normally when you see these in silico or in vitro studies, these are often run side by side. Now, in a this rational drug discovery process, what happens? The biological target plays a vital role in this rational drug discovery process, and the physiochemical understanding of the biological target can lead to optimize the drug candidate. And this biological target is often characterized with the help of some analytical tools like NMR, you can say X-ray diffraction, that biological tools, analytical tools, and this biological databases are often utilized for the development of the selective and the specific drug for the particular disease. Now, this is a change that occurred after the computational, or you can say that the chemoinformatics knowledge. In earlier stages, what happened? The drug candidate are randomly discovered. Means what? And after discovered, and after that discovery of the drug candidate, the mode of action has been identified. But nowadays, we can modulate, or we can develop a molecule with the predetermined mode of action. Means we can selectively go for a particular enzyme for the targeting, or we can selectively go for inhibitor of that particular enzyme in a cancer biological process. So, due to this chemoinformatical knowledge, or due to this development in the databases, this biological target knowledge have been improved, and that and improvement in this biological knowledge, the drug candidate or the development of drug candidate or drug discovery process have been tremendously improved, and the speed of that drug discovery process has been improved now, improved in a quite a large way. And the ultimately, what is the fundamental output of this in silico modeling and in silico development of the drug? That is a rejection ratio has been minimized. We can't say that there will be the zero rejection ratio, but the rejection ratio can be minimized, because in earlier stages, the ADMT prediction has been carried out after the animal studies and all those things. But in due to the advancement in this computational tools, the ADMET study can be carried out in early stages, so that the molecules which does not fit in the desired uh, uh, criteria of the toxicity or ADME behavior can be straightwise rejected, and the optima, or only the molecules which are fitting into the bracket of this ADMET, or you can say that toxicity bracket, that can be these can be further selected for the further optimization. So that's why the overall cost effectiveness has been achieved. Also, the drug discovery process, speed of drug discovery has been improved. So that is the impact of this chemoinformatics on all over pharmaceutical research, or we can say that in a or in a drug development processes. So this chemoinformatic due to this chemoinformatical improvement in the chemoinformatic processes, the rapid development of the drug takes place nowadays. You can say you can easily know you can easily uh, easily can search the number of drugs which are coming in the market nowadays. For example, bedaquiline, that is a anti-tubercular drug which is discovered after 15 years of the previously discovered anti-tubercular agent. So, due to with this drug discovery process, this speed can in silico process, this speed can be minimized, and the toxicity effect which is associated with, or toxicities which are associated with the developed drug, or with the developing drug, can be minimized with the help of the chemoinformatical purpose. Now, I think I will skip this slide, the overall process. Now, what happens in drug discovery of this 21st century? Now, here we can say that these computation methods are used to select the subset that is to be tested in the lab based on the prediction of drug likeness, solubility, binding, that we have started to discuss the kinetic and toxicity and side effects. And this will minimize the overall experimental load, because these computational tools or computational technologies are highly, we can say, that are efficient, so that the number of the molecules which are developed, that can be minimized with the help of these computational tools, and which can reduces overall experimental load which are normally present in the lab for the development processes. Now, when you see a major aspect of this chemoinformatics, what is the exactly chemoinformatics has been combined with? The chemoinformatics have been distributed into three different parts. That is the experimental data, that model generation and prediction of the unknown compound. Now, why these three are the pillars of this chemoinformatics? Because this chemoinformatics is often nothing but the transforming chemical knowledge, or chemical know-how into the computational language. For transformation of this chemical information, we require a large experimental data, large true experimental data, so that the models, or the predictive models which are generated, can be generated, and these models can be used for the prediction of the unknown compound. Now, with the uh, continuous research, and you can say that continuously the molecules have been developed and the data has been proven. So we got a large data. That data can be processed with the help of this chemoinformatics, and the models can be generated, and these models can be used for the minimization of the upcoming molecular number, of minimization of the upcoming molecular number. For example, when you see when you see an example for that, when a new beta blocker comes in the market, and one person has to be want to develop HPLS method or any method for that new beta blocker. Now we got a major chemical structures or major class of beta blockers which are already developed, and we got a mobile phase composition for the estimation of these beta blockers. So simply we can correlate the physiochemical properties of that beta blockers with the with the retention factors, and we can identify the mobile phase composition by uh, that simple model generation. So that is output of this chemoinformatics. So that we must have a good experimental data. With the help of this good experimental data, the model can be generated, and these model can be utilized for the prediction of the unknown compounds. Now, going to deeper of that model data collection on all these parameters, the information acquisition and management is the primer of the chemoinformatics tools. Means what? The method for collecting data, that mainly experimental development of the database for storage and retrieval of the information. Means what? The information acquisition. Normally, the number of quantitative QSAR, or you can say QSPR models, or you can say the QSRR model, are fails in this part, because the data which they are handling. Normally, there are not a fixed rules, but the in a QSAR or in any qualitative parameter with this computational tool, you must require a data which has a maximum coverage of the all the chemical space. So the collection of the data is a major issue in the uh, computational tools. So if you got a right data, then you can easily develop a good model, and this model can be used for the development of the or the predictor of prediction of the unknown compounds. So that is the uh, basic fundamentals of this human parameter, that is the information acquisition and management. So data selection, it must be carried out very carefully. Now after that, the information used, that is the data analysis and correlation and model building. Now this is nothing but the application part, that is how the data can be handled, which types of method can be used for the handling of that particular data, which type of models can be used. So selection of that models or the statistical understanding of that particular models is depend upon the person who are using that particular information. So this comes after the continuous experimentation or literature review, or continuous exposure to the model, that data handling. So this is nothing but this is that nothing but the expertise domain of that particular person. So that person should be expertise how it will be correlating the system, which types of model he can be used, or which can types of technology that person has been continuously using for that handling of that particular data that comes into the picture. Now after that, the application means the predictive behavior of that particular system. Now once we have got the data, then we have developed model. Now how that model can be used for the prediction of the unknown system. So that is the application part of the chemoinformatic tool or your computational tool. Now this information or prediction behavior, this prediction should be correlative. Means we can't predict a beta blocker system with the model which are generated with the morphine like molecules. So there should be some similarity, that there should be some relevanceness between uh, the you can say that you can say in our term, that is the training set and the test set also, or the external test set also. So that the model which is generated and the predictive behavior should be run in a similar line. Means what? You can say that we can't correlate, or we can predict a generalized system for a six-member ring to the five-member ring system. So there should be some correlation between the you can say data acquisition and the predictive behavior, or the data which is or the data which is handled, and the unknown compound that should be the behavior of that unknown compound that should be predicted with the help of that data. So that is a third most the third part of that your chemoinformatics technology, that is the predictive behavior. Now when you see that chemoinformatics technology, the often terminology comes that a computer simulation. Now when you see a simulation, what is exactly a simulation? Now by the definition, a simulation is a process to create an imitation of a real world system or a physical object into a computer model. It is a process to create a imitation of the real world system or a physical object to a computation model. And this simulation is is a nothing but the representation of the real world system. A simulation should be a representation of the real world system. Normally, how this simulation can carried out? Now, this simulation can be carried out with the performing experiment to understand behavior of the system and evaluating the new strategy. Simulation is carried out and then observing the event, process, and properties and the behavior of system with the computer model. All this technology comes under the computation. Simulation means nothing. Simulation doesn't stop over the development of the model. Now normally when you see in earlier stages in last before 15 or 15 to 20 years back, the simulation only stopped at the development of the ball and stick model of any system, that is the imitation of the real world system, how the molecule looks like. Means you often find the benzene when a ball and stick model comes, when a stick model comes, how it is look. That is normally previously it is called as a simulation of that particular benzene in a computer you can say in silico way. But now the simulation is far more advanced than the only imitation of the model of that real world system. But it also includes the performing experiment on that developed model or developed system to understand the behavior of that develop a model, and this behavior can be used for the development of the new strategies. Means what? Simulation doesn't stop at the development of a re-imitation of the real world system, but also it involves the performing experiment to understand the behavior of the system and the evaluation of this new strategy. And also this simulation includes the observing the total behavior, processes, properties with and correlation with the real world system. So it is nothing but the development of the model, performing experiment on that system, and the correlating the output of the developed model with the real world system. So based on this simulation system, we can say that the newer computational tools have been developed. Normally, when you see a system versus its model, what we find, a real system and the model is nothing but the model is a simplification, or you can say that the abstraction or assumption which is associated with this real world system. This is the basic of the simulatory system which are nowadays which happen. Means what? In a simplification, or we can say that we can develop a protein model with of n number of amino acid in within in that system with a computational model. So that is a simplified model of the real world system. When you see a protein model, when we see a protein structure, primary, secondary, tertiary, quaternary structure, it is a quite complicated one. We can call it as that the primary is amino acid sequence, and secondary beta sheet, alpha helix. But with the computer model, we can easily observe the primary sequence, the list of amino acid, the beta helix, then beta sheets and alpha helix, then the cavity which is present in the protein can be easily understood with the help of the model, because we can't say a common layman cannot understand what is the cavity. Okay, cavity in the active site, or cavity in the protein, or active site in the protein. But when the protein have been observed in case of the computational knowledge or computational tool, we can easily observe that the curvature or clip which is present on the protein cell can be called as a cavity, or the pocket in which the native ligand binds, it is called as a cavity. So how this cavity look like? What are the fundamental amino acid which are present in that cavity, or what are the distance between that amino acid, or distance of that amino acid with the each other, and how the molecule can be designed. So that is only achieved with the help of the computer simulation, or you can say the chemoinformatical tools, so that the real world can be simplified or abstracted, or we can say that, or it is correlated with the uh, modular system, so it can give a basic understanding of the all the system. Also it can improve the uh, overall output of the research, or it can speed up the process. Now when you think, if a can say, if a binding pocket model is not available with the computer system, how one can develop a molecule for that particular system? He try a number of molecules. Suppose he want to try develop a inhibitor for that molecule. He can synthesize a number of molecule, test that molecule with the biological assay methods, or you can say animal tested systems, and after that uh, the activity will be predicting. So that is a overall flow, which is, if a model is not available, if a model is available, he can develop the you can say that the uh, chemical model of that particular drug, or you can set an inhibitor, try to figure out whether it is binding with the protein model or not. If it is binding, then it will be analyzed for the ADMET and toxicological behavior. If it has an excellent ADMET toxicological behavior, then it can be used for the further develop. It can be used for the uh, further optimization process. So that is a journey from a real world to the modular system, and how that, and this, by this way, the modular system can improve the overall understanding of the protein systems, or overall understanding of the biological process, or overall understanding of the drug discovery processes. Now, how the simulation can be achieved, or how that simulation can be carried out? When you see the overall flow of the simulation, which is associated with the system. Now, for simulation, we must have a system, and the person should define a system in a particular way. Means what exactly the system is all about. If we are suppose the simulation of a protein is taken place, then he has to define how many amino acids are there, what are the total number of chains which are present in that amino acid in that protein, how they are linked, what are the probable sites of the disulfide linkage, and all those things, all those steps all together. So that definition of the system includes all the basic information of that particular system. After that, the model or the basic model of that particular system has been developed. Then the data which is associated with that system will be collected, and then the model will be translated to the computational tool or computational language, and then verification of that model with respect to the basic input or basic requirement of that system will be carried out, and then exact last experimental will be derived, and then the model will be uh, model will be documented. That is the process of a simulation of that particular system from real to a modular system. Now, after that, we'll be discussing about the basic tools which are used for this pharmaceutical research. Now, in my discussion with the Honorable Dr. Choudhary sir said about the QSR and QSPR. So we'll be talking about two tools, that is QSR and molecular docking, into this session. Now, this QSR first. So before to start, we start of this QSR session, I want to discuss with something interesting with you, that what are the fundamental modeling strategies, what are how the strategies can be selected. Now when you see when in a computational chemistry point of view, or medicinal chemistry point of view, we often deal with the two system, ligand and receptor, means target and drug, or ligand and target. So when you see when we define in the modeling strategy in terms of these two counterparts, or these two systems which are playing important role in the overall drug discovery process, this receptor system has been classified into two different parts, that is the unknown and known. Now, how it is unknown and known? Now, unknown and known is a terminology which is associated with the knowledge of the overall structure of that receptor, not about the name of that particular receptor or role of that particular receptor. Now, why we are talking about this unknown and known? Because in a human body, there are a number of proteins or a number of receptors which are present in a human body, and normally there are different types of receptor like G protein coupled receptor, iron gated receptor, nuclear receptor. Now, this location of that particular receptor often uh, often describes the overall structure of that particular receptor. Now, if a structure of the particular receptor is known, means what total amino acids, the number of amino acid binding cavity, all is already well documented, then we can easily take up that receptor. You have already known the cavity binding cavity, you have already known the amino acid which are playing key interaction with the receptor, or you can say that in the in the binding analysis, you can develop your own set of molecule with the help of that known, not unknown receptor. If the structure is unknown of that receptor, structure is unknown of that is not known, means we only known that know that this is a GABA receptor which is commonly used for the which is playing vital role in case of the CNS. So if a GABA receptor structure is not known, means structure in case when we define the structure in case of modeling, we only often deals with the 3D structure of that particular receptor, not the amino acid sequence. Only the amino acid sequence is known, but 3D structure is not at all available. Then one how one can go for that particular receptor. So here comes the terminology that is a homology modeling, that one can develop its own structure with the help of some modeling tools like Swiss modeler and all those things, and uh, these structures can be used for the uh, further drug development process. Now, he that is a two concept which is associated with the receptor. Here another part is there that is known as the ligand, which plays important role as to that of receptor. Now, if a ligand structures are unknown, means what? The native structures which are have capability to bind with that particular receptor are totally unknown. Then how that molecule will be developed? So here the combinatorial technology, or HTA6 commonly used, means you have to hit a number of molecule with that particular receptor structure, and uh, based on the heat analysis, you can identify the basic molecule, the basic structures which are have capability to bind with that particular receptor. You can optimize that molecules, and you can develop your own set of ligand over there. If your ligand structures are known, then here comes the technology which is commonly nowadays is upcoming, that is QSR, which is commonly used for the formulation technology and analysis that we have already discussed. That is a 3D 2D QS, 2D 3D QSR and pharmacomodeling modeling. Here, what happens if the ligand structures are known, their properties will be known, and the properties will be correlated with the biological activity, and the important properties which are responsible for that particular biological activity will be identified, and these properties will be optimized or will be used for the or this can identify and optimize, and this which can improve the overall biological activity or overall behavior of that particular molecule. That is the basic fundamental of this QSR analysis. If ligand structure is known, your receptor structure is known, directly go for docking, means you have to directly go for the binding analysis, which part of the molecule is binding with that particular receptor, optimize that part, develop your own set of molecule, and you can get a potent molecules over there. Now, let's begin with the second part of my discussion, that is the tools which are commonly used. Now, this is first tool, that is QSR. Normally, when you define a QSR, it's everybody knows that this is a pioneer work of the Sir Crowning Hens. Now, this Hens define QSR with the help of a simple one-line statement that a structure of a chemical influences properties and its biological activity. Means what? Structure of a chemical influence its properties and ultimately the biological activity. Now, in the start of the our session, we discuss about how the properties can be correlated with the interactive behavior, and interactive behavior can be correlated with the biological activity. The hence thought in a similar way that in similar way that the structure of a chemical is often influence the physiochemical properties, and these physiochemical properties often influence the biological activity. And he also thought that the similar compound have a similar have can behave similarly, or similar compound can act a similar way, because these compound have similar physiochemical properties, and the similar physical chemical property can dictate the similar biological activity. Based on all this assumption of this crowning, hence the QSR technology have been or QSR tool have been developed. Normally, when you see a QSR as a detail, the normally what happens in a QSR when you see when you see physical properties of the drug molecule, in this physical property of drug in part dictate their biological activity. Means what? The physical property, the physical behavior of the molecule often be used for the partitioning of the drug molecule, often be used in the release of the drug molecule, or often be utilizing uh, in the target specificity, or you can say that the molecule can reach, or it is the major parameter which decides the whether a molecule reaches to a site of action or not, or it is get absorbed or not. Means what? This physical, because this physical properties is often be utilized in the overcoming of the physical barrier which are present in the human body. Normally, when you say in the human body, so here this QSR has been evolved, and QSR is nothing but a statistical approach to use these properties, which are nothing but the physical properties, in the development of the mathematical model that will relate the physical properties to the biological activity, and shows how those mathematical models can be used to understand drug action and the drug design purpose. Means what? If a physical properties of the drug are detecting their ability or detecting the biological activity, and QSAR is utilizing these properties to develop a statistical model to understand how these properties can be useful in the drug discovery process, or in the overall drug development process. Normally, when you see a QSR as a whole, QSR is nothing but a mathematical relationship between the biological activity and the molecular system, and its geometrical and chemical characteristics. You can say that it is nothing but a mathematical, you can say linear relationship, not usually a linear relationship observed over there, but it is nothing but the mathematical relationship, or we can say that it is a simple mathematical equation which correlate your property with the physiochemical properties of that particular system, or of particular uh, molecular system. And these mathematical relationships is often utilized to develop further drug molecule, or often utilized in the drug discovery process. And with the solution of this model, we can easily understood which particular properties are significant to our biological activity, and which properties are not significant, or you can say insignificant for the biological activity. And these properties can be modulated to develop a potent or selective, definitely specific drug molecule. Now, normally in a QSR, there is QSR is normally used, or it tries to find a consistent relationship between the biological system and the molecular properties. Means what? There in the QSR, a consistent relationship is commonly observed between a biological property and the biological activity and molecular property. Normally, when you see this, why this consistent relationship? Because these rules are can be used to evaluate the activity of the new compound. Means to attain a high grade of, you can say that a high amount of predictability, or to attain, or to have high levels of predictability, the consistent relationship between the biological activity and the molecular property has to be there. So QSR identify these consistent relationship and show that this QSR has a high degree of predictability. The QSR model often has a high degree of predictability, and it always establish a equation of relationship between the properties and biological activity. You often find that a you means you doesn't find a QSR model which it does not have any relation model which is model doesn't find a QSR model which does not have any relationship between the properties and the biological activity. In a QSR, the consistent relationship between properties and biological activity is often find, and this relationship is commonly used for the predictive behavior, or prediction of the biological activity of the new compounds. Now, when you see overall behavior of the overall flow of the QSR work, normally what happens? What is the exact change which is occurring in case of the QSR and normal drug development process? Is often that is a change in the overall flow. You know, in a common routine drug discovery process, what happens? A molecular structure will be developed, will be synthesized, and the activities will be calculated. But here, in a QSR way, the molecular structure will be developed, but in silico, their molecular representation, which is simulation based. Then the properties will be explained in case of descriptors, and the descriptor will be correlated with the biological activity which is already known, and and then the model will be generated and the prediction of the unknown compound will be there. So this is overall flow of the QSR work. Normally, when you see a QSR model, how the QSR model is normally look like? Normally, the problem of QSR is to find a relationship between the biological activity and the physiochemical property. Normally, we find any QSR model can be given in a simple way that biological activity is equal to C0 plus C1P1 into Cn into Pn, means that is the correlation between the properties and the biological activity. We often find, or we doesn't find any QSR model which has a molecular structure in it or all those things. It is the only correlation between the property and the biological activity, and the prediction error is minimized because a number of molecules can be used for the QSR model. QSR normally use a huge number of molecules so that the predictive behavior will be more, and in a normally there are certain rules that we you there are certain rules that which they are not written rules, but we often find for a single property that we consider on the QSR, you should have five molecules in the data set. So ultimately, what happens with the huge number of data sets, a huge number of molecules represented in data set, the overall coverage of the property or space of the property, chemical space will be more, and the predictability ultimately increases in case of the QSR models. Normally, why when we require a QSR, or when you see applicable domain and all those things, a need of QSR is defined by this simple statement that number of molecules required to synthesize in order to place 10 different groups in a 4 position of benzene is equal to 10 raised to 4. So simple mathematical calculation is there. If you have to place 10 different groups in a four positions of benzene ring, the molecule number becomes 10 raised to 4. If the, in that way we have to synthesize a huge number of molecules which require huge efforts, then the biological activity will be more. After that we will be getting the actives and inactive or intermediate active molecules. But with the help of the QSR we can minimize all these processes. We can synthesize a small number of molecules, correlate the small number of molecules, test that molecule with the biological assay methods, develop a model to correlate the molecule with the physiochemical property, and after the QSR model development, predict the remaining molecules with the help of the QSR model, and only and select those molecules which has excellent biological activity in the or excellent biological activity in the QSR models, or the models which are generated with the help of this QSR system, and those molecules will be further optimized. So ultimately, in a by the QSR way, or we can say that in the modern way, we can minimize the overall experimental process, manual efforts, or we can say that the laboratory workload can be minimized. Now, why one go for a QSR? What is the rationale behind the QSR studies? Now, when you see a drug design process, in vitro potency, in a drug design, in vitro potency only addresses the part of the need. Means biological assay by in vitro only addresses the only small part of the need. Means what? This in vitro potency doesn't describe the overall molecular passage or molecular path to the reach up to the target, up to the biological target, or you can say that it does not include the ADME, that is absorption, or all those parameters cannot be predicted with the in vitro potency. So here, this due to the addressing the only part of the need, a successful drug must able to reach its target while still in active form. So due to this important admit behavior, or you can say the importance of this admit, that other kinetic behavior of the molecule in the biological activity process, we must have a system which can be which can predict both the parts of that is the kinetic also and dynamic also. So that's why the QSR has been evolved. Now, when we see in vivo activity of a substance is a composite of many factors that everyone knows that, including intrinsic reactivity of drugs, solubility and physiochemical properties, or ability to cross BBB. It's non-reactivity, not with the non-target material, that is steric behavior. So, in a QSR manner or in a QSR technology, we can correlate the measurable, calculable, physical or molecular property to the same some physical, some specific biology in terms of an equation. Means what? In a QSR in a QSR way, we can only we can correlate the dynamic contribution also and also kinetic contribution of that molecular system towards the biological activity. But think upon the docking, that another tool, but docking only deals with the interactive behavior, the contribution towards the dynamic part. It does not include the contribution towards the kinetic part of the system. So that's why nowadays the QSR have been evolved, and QSR given us so much emphasis. Nowadays, you often find a newer types of QSR, 4D, 6D, 7D types of QSR. Then the another technology that is developed by the V Life Sciences is GQSR, group-based QSR. You often find HQSR, COMSA. These are the all are of the QSR, means QSR technologies which can be used for the development of the drug discovery process, or they can be used for the other application domain like QSARR and QSPR also. And once a valid QSR equation can be generated, it should be possible to predict the biological activity of related sub-candidate before they are put into expensive time-consuming biological testing. That we have already discussed that. It can minimize the experimental load and all those things. Now, so after the discussion of the rationale for the QSR, now how one can select the data means, now in this start of our discussion, we discussed about the importance of the correct data, how data can be uh, more can lead to the precise results, or you can say that the good chemoinformatical approaches. Now, QSR is so success of the QSR is often depend upon the data that is used for the development of the QSR model, because QSR is nothing but the relationship between properties of the molecules in the data set and the biological activity of that particular molecule. So success of the QSR, development of a good QSR equation or successful QSR equation is solely depend upon the quality of the data which is present in the or quality of the data or the quality of the molecule which are present in that particular data. So there are some prerequisites for this QSR data, that how the data can be selected. Now, that first is all analogs should belong to congeneric series. Now, this is quite change in the development of the QSR process, because you know earlier traditional QSR process, this is a strict regulation, or it is a uh, the prime most important feature for the data set, because the all analogs should belong to continuous series. Means what, they should have a same structural pattern, or they should belong to same uh, series of biological same series of chemical species, so that the biological so that the physiochemical property should be similar, and they should be having similar behavior in the biological process. So this congenericity, this often you can find in a 2D QSR or traditional QSR way. But nowadays, when you find another advances in the QSR, that 3D QSR, 4D, where the congenicity is not regulated, that is, you can say that the non-congenic QSR can be there. But when we go for a traditional 2D QSR methodology, that is common physiochemical property with the biological activity, then the congenicity should be there, because these physicochemical properties are solely depend upon the chemical behavior of chemical structure or the structural pattern of the system, and this structural pattern can be correlated pattern of the system. And this, if a structure pattern change, there will be a huge change in the physicochemical properties, and so that can be observed in case of the QSR, or that can lead to the false QSR model generation. So this is the first regulation, that all analogs will have should have same mechanism of action. Means what, although they are belonging to congeneric series, but they should be have same mechanism of action. Means if they're having if we are going for a anti-hypertensive QSR. We are calling it. We got a molecule from anti-hypertensive class, so we can't go for a single data set which has a calcium channel blocker, beta blocker, AC inhibitor, or you can say that certain class of molecules, because all these have different set of mechanism of action. Also, due to their mechanistic changes, their structure requirement is changed, because every receptor is have some different binding cavities, binding domain, or you can say that the binding active site, so that their structural requirements are quite different. So their structure requirement, due to their structure require change in the structure requirement, their physicochemical properties will be changing, or there will be different physicochemical requirement for that particular uh, receptor. So all analogs should have same mechanism. So means what? If a biological activity is same, but their mechanism of action is not same, then we can't go for a same sort of molecule in a singular data set, because which may lead to the false types of QSR model generation. Also, another requirement, all analogs should bind in a similar fashion, which commonly observed in case of the allosteric inhibitor. Means what? Suppose we are going one researcher working on a QSR of the uh, HIV reverse transcriptase inhibitor. Okay, so we got a reverse activity of antiviral, which is similar. All molecules are congeneric, also similar, but one is nucleoside reverse transcriptase inhibitor class of molecule, another are non-nucleoside reverse transcriptase inhibitor, nevirapid class of molecule. So when you see when you see chemically the nevirapid class of molecule and zoludine class of molecule, both are having similar biological action. Okay, they are having antiviral action, mechanism action is also same, reverse transcriptase inhibitor, but their mode is different. One is allosteric inhibitor, another is active site inhibitor, and due to their change in the biological action mode, their structural why there is a change in the uh, change in the biological action mode, because their structures are quite different. One is a, you can say, the pyrimidine class of molecule, another is a heterocyclic system, nitrogen sulfur containing system. So their activity pattern is same, mechanism of action is same, but binding is different, and their physiochemical properties are also different. Physiochemical properties are different, so we can't put molecules of these classes into the same data set, because which may lead to the false QSR model generation, and which lead to the false predictive ability. So another is the effect of the isostatic replacement can be predicted. Means what? We can put a isostatic replacement or molecule which has a isostatic replacement in a singular data. Means C double bond O can be replaced with, if a molecule which is replaced with the C double bond N, which can be considered into the singular data. Also, the binding affinity can be correlated with the interactive energy, or it can be correlated with the interaction energy. That is, the ionic effects, or means ionic effect can be made approximately constant. The biological activity can be correlated with the binding affinity, means the ability of the molecule to interact with the receptor. But here, the transport properties cannot be correlated, or not with the transport properties. Transport properties are quite different to biological activity. Biological activity is when you see interactive behavior is there. Also, in the transport properties, the interactive behavior is there, but they are not considered over here. The biological activity is correlated with binding affinity. Binding affinity is correlated with the interaction energy. Means what? We can say that the interaction energy, or you can say interactive behavior of the molecule, can be correlated to biological activity with the help of the structural behavior, because the interactive behavior is a responsibility, or it is a contribution of the molecular structure, or you can say chemical structure of the system, and which can be correlated with the biological activity, or the biological activity of the system. So it is all about the prerequisites of the uh, QSR study. Now, uh, in a QSR model generation, we often observe a terminology which is called as a descriptors. Now, what are descriptors? Now, descriptors is nothing but the mathematical expression of the biological physiochemical properties, and they are utilized to relate structure to its activity. They are utilized to relate structure to its activity. Normally, when you see a descriptor, they acts as the independent variables. They act as the independent variables, because when you see an overall QSR model, we got a two variable over there, independent and dependent. Now, biological activity is dependent upon the physiochemical property, but the physiochemical properties are not dependent upon the biological activity. So, The representation of the biological activity, physiochemical properties are nothing but the descriptors. Then they can be actually independent variable, because why we require descriptors? Now one have to define lipophilicity of the molecule. How one can define lipophilicity? Because it is lipophilicity is the property of that particular molecule, but we can't have a structure, or we can't have mathematical expression for the lipophilicity. That's why we require log p, log s, such a physiochemical property which is a representation of the overall lipophilicity of the system in terms of the descriptor, or in terms of the independent variable, which is mathematical expression of the physiochemical properties. Now the descriptor can describe different aspect of the molecule in a single way. They can compare a different molecular structure. They can compare a different conformation of the same molecule. That's why we require a terminology which is known as a descriptor, which is the mathematical expression of the physiochemical properties of that particular system. Now, after that, let's begin with the typical workflow of the QSR study. How QSR studies can be carried out. Normally, when you see a QSR, start with the data selection, then descriptor evaluation, then distribution of the training and test set. Normally, we often find in automated modes of the QSR model generation, you often find there are you can have manual data set selection also. Means how much percentage of the data you have to select for the uh, as a training set, as a test set. Means normally 70-30 or 80-20 percentile distribution are there. Now there are certain advanced softwares are there which also distribute the test set into external test set and the internal test set. Means what? We have 70%, 20% and 10%, that is as external test set. Then variable selection, which types of variable, statistical evaluation, model interpretation, and after the interpretation, development of the lead structures. Now there are different types of QSR which are commonly observed or routinely observed in the scientific literature, that 1D QSR, 2D, 3D, 4D, 5D, 6D, and GQSR. Now, this is a typical 2D QSR that is we have calculated with our software, that is V Life Sciences. Now, this QSR, 2D QSR is one fundamental QSR, that is a typical QSR methodology, and it is alignment independent, because 2D QSR only predict the physiochemical properties of the biological activity. It doesn't predict the interactive behavior. So how the molecule is interacting with the target site or the receptor cannot be predicted with the help of. So here the alignment cannot be there, or alignment cannot be carried out, or for that alignment of the molecule is not required. Now, what are the different 2D descriptors? I have enlisted n number of descriptors, that is physiochemical descriptor, Wagner index, functional property group, eigenvalue, connectivity, and all those things over here. And what are the different methods of this QSR, 2D QSR? That is the quantitative regression techniques, pattern recognition, Hammett relationship, simple linear regression, PLS, PCA, that is the principal component analysis. That now next comes the 3D QSR, which is nothing but the three-dimensional QSR, which can predict the interactive behavior of the molecule under study. Here, the alignment of the molecule, a prerequisite is only that alignment should be there. Alignment of the molecule should be carried out. To 3D QSR, normally two different types of fields are considered. The study can yield static field, because in a interactive behavior, or in a interaction of the molecule with respect to receptor, lipophilicity does not play any penultimate role over there. So these lipophilic parameters are not considered in a 3D QSR. Only the steric and electrostatic properties are considered. Now, how the calculation of the descriptor? Now, normally the descriptors in a 3D QSR are quite different to that of 2D QSR. In a 2D QSR, we got a physiochemical properties over there, but in 3D QSR, they are often calculated as hydrophilic, steric, and electrostatic interaction energy at the lattice point of the grid. That is these are the nothing but the interactive energy that are required for the interaction of the molecule with respect to receptor, and they are point of the grid using a methyl probe charge 1. This field provides a description of how each molecule will tend to bind with the active site. Means the only this predict the interactive behavior, and these fields are required to provide exact description of the molecule, which have ability exact description of how each molecule can be having binding capability with the active site. Normally, these field descriptors are typically consist of a sum of one or more such special properties such as steric factors and electrostatic potential. Now, that this is another technology that we have to I want to discuss with you. That is a QSA, GQSA, that is group-based QSA. Now, what happens in this GQSR? In this method, every molecule of a data set is considered as a set of fragments. It's also called as a fragment-based QSR, and the fragmentation scheme either a template-based or user-defined. Means what? You can fragment a molecule as per template-based. Normally, when you say a generic series, we can easily identify where the common substitutions are there. We can take that as a fragmenting origin, or we can say template, and based on that fragment we can distribute molecule into a number of fragmented sets, and these after that the descriptor will be calculated with the normal technology convention, that technology, and they are correlated. Now, what is the change between a conventional and GQSR? Normally, in a GQSR, make the computational chemist or the person who are working get a critically important site-specific clues within a molecule where a particular descriptor can be modified. Means what? When you see a conventional QSR, we often find that this part of the molecule, lipophilicity, should be addressed, but lipophilicity of which substitution, it cannot be addressed in case of the typical QSR, which can be addressed in case of the GQSR. Normally, when you look at the molecule over here, that it's our own publication from one of the reputed journal, where we identified 7 fragments from a data set, and these molecules are fragmented based on these 7 fragments where the substitutions are commonly there, and these 7 fragments are the molecules are fragmented to 7 fragments, and their physiochemical properties are calculated. We often find, look at them, and then they are correlated to biological activity. We observe that look at the R7 volume. Volume is a positive contribution. Means we have to consider the volume at the R7 substitution only. Means we have to go for a molecule which has a bulkier, more sterically important groups, which can improve the biological activity. And then, similarly in a positive R2, that is which is indicated by the alphabet I over there, which has a S log lipophilicity contribution, which is also positive. So that lipophilicity contribution can be considered only in that substitution. In conventional QSR, this peculiarity is not there or cannot be observed in case of the conventional QSR. Now, after discussion of this, how this QSR model can be validated, what are the different validation pattern of this QSR model, how they are validated. Normally, they are validated with the help of the statistical quality, that the regression coefficient, then predictive coefficient, outlier, and predictive reliability of external set. Now, this all first three are the uh, statistical parameter, but the last is the experimental parameter, that is the prediction reliability of the external set. Means we can have R square of greater than 0.8, we can fit that values. But the prediction reliability should be there, because normally it is thought that if a QSR model not have good predictability, then that model is not of use. For that, for the development of the QSR model, normally when you see good softwares or good math, good technique or good uh, computational tools often use two types of validation, that the internal validation, also external validation. Now, what happens in the internal validation? The data set of the molecules are distributed into test, training, and from the test, certain molecule and that test set is used for the internal validation. Means what happens in the internal validation? Normally, if a QSR model is not able to predict the molecules in the data set, then that model is not of use. That is the overall consideration over there. Means the model should have good predictability only when will be having good predictability only when it successfully predict the the biological activity of the internal data set. If it is predicting or it has capability to prediction of the internal data set, then it can be used for the external data set also. So that probability reliability is the most important criteria for the validation of the QSR models. What are the advantage when we see a QSR as a tool? The quantifying the relationship between a structure property can give the understanding of the effect of the structure with the activity that can be carried on. Then it can be used to predict, make a potential decisions, and it can be used for the understand between the which are the functional groups which can be used, which are utilized for the molecules with the greatest activity and with the target. So with with the QSAR, we can correlate in a single tool. We can correlate dynamic behavior also and kinetic behavior also. It cannot be achieved with the other tools also. So this is the most important tools. Normally, what are the disadvantages which are associated with this QSR? If it as a tool, it has certain advantage as a certain disadvantage. The fundamental disadvantage is false correlation. Means false correlation is can arise, and because why? What is the fundamentals? Why this false correlation will be there? Because this QSR is relied too heavily on the biological data, and by its nature, it is biological data has considerable experimental error. Means QSR is solely depend upon, or when you see a QSR, we often find it is depend upon the biological data. Means biological activity is correlated with the physiochemical properties. If biological activity is faulty, or we can say faulty, but by its nature, it has a considerable variation, means species variation. Then we can say that expected timing variation, then tool variation are there, because biological activity has by its nature subject to considerable experimental errors. So this due to this heavily rely on the false then biological data, it might lead to certain false correlation. Also, frequently experimental upon which QSR analysis depends lacks in the strict sense of experimental design. Therefore, the data collected may not reflect the complete property space. Means the data which is collected does not a reflection of the complete chemical property space or space which is associated with the molecular system. And many QSR results cannot be used confidently because this uh, due to the lacuna of the complete property space, or the lacuna of the overall chemical properties. So we can't get a good predictability over there. Also, whereas physiochemical properties are known to be cross-correlated, means what? The continuously same types of uh, parameters if comes into a single model, then it's called as a cross correlation. So for a for a good predictive model, or for a good set of experimental design, a QSR model should have reflection of the all three types of physiochemical property space. Means what? It should have some lipophilic parameter, it should have some steric parameter, it should have some electronic parameter, because the biological activity is often contribution of all these three parameters. So if a cross correlation are there, this is a major disadvantage which is associated with QSR. Normally, when you see a QSR as a success story, there are certain reasons for the failure of the QSR. First is the false correlation that we have exhaustively discussed. Then may not reflect the complete property space. Bottom line, that is overfitting of the, and in last is the that is the based on the computational chemistry, that is inability to interpret the QSR model. That is the most important thing. That is the inability, that is improper interpretation of the QSR model may lead to the failure of the QSR studies. Okay, so hello, sorry sir. Hello, hello.
Yes,
Hello.
sir. Ah,
Yes.
madam. Continue.
Shall I continue the
Uh-huh.
talking, or
Sir, you have more 5 minutes. You may continue
okay,
for
okay,
5 minutes, or we will have the question
okay,
answer in 5.
uh, okay. Then I can stop here only. So uh, because
Okay.
in 5 minutes the talking cannot be completed. So
Okay. Okay. Okay.
okay.
Kulkarni madam.
So
Oh,
thank you very much for
hello.
the hello. Yes,
Yeah, we can go for question answer session for a
yes.
couple of minutes if the participants have.
Okay.
Participants, if you have the questions, please ask. Hello. Any questions from participant side? If there are no questions, I would request Dr. Choudhary sir to take the photograph, e-photograph of today's event. May I request all participants to on the video? Choudhary sir?
Yes, madam. Yes, बोला
हा
madam.
घेतला photograph?
नाही, एकच मिनिट हा, एक मिनिट.
Ha, please. Sure. I can see smiling Sadhna. Hi.
Very good morning.
Yes, madam, घेतलाय.
Thank you so much. An
Yes, madam,
organizing
continue.
committee of DTE sponsored faculty development. I sincerely thank Dr. Praful Choudhary sir for his highly enlightening speech and a great presentation. We all appreciate your convincing speech on computational tools for pharmaceutical research, sir. Thank you so much, Dr. Prafulla, for making the QSAR so much interesting and simple by your own developed presenting skills. May I please request Dr. K.B. Burade, sir, Principal Government College of Pharmacy, Karan, to present.
गोंजारी सर, मॅडम बहुतेक लॉग आउट झाले.
Yeah. So,
प्रफुल sir, please accept this certificate, virtual certificate.
Okay. Thank you, sir. Thank you very much.
See you. Thank you. Thank you very much.
Okay, sir. Thank you.
yeah, thank you all the participants. And our next session will begin soon. So we
The
will
next
have.
speaker is already joined.
Shall we continue,
Hello.
sir?
Yes, yes. We'll continue.
Okay. Our next session. Our next session is on basics and application of UV visible spectroscopy. And the eminent speaker for this session is Mr. R. Girijan. I would request Professor A.S. Kulkarni Madam to take over the session. Thank you.
Thank you, Gonzari sir. Good afternoon, dear participants. It's a great privilege to introduce you to the speaker of today's second session, Mr. R. Girijan sir, who is a renowned personality in the field of analytical chemistry. Mr. Girijan sir is a product consultant in the field of analytical instrumentation with around 44 years of experience in the analytical field. He is a postgraduate in analytical chemistry. He is fellow of Institution of Chemists, India, member of American Chemical Society, and life member of Indian Society of Industrial Chemistry. Sir has worked in pharmaceutical as well as paints and polymer fields and has approval from FDA Maharashtra in the field of chemical and instrumentation analysis. Sir has extensive experience in chromatographic and instrumentation, chromatographic and spectroscopic analysis. R. Girijan sir he has delivered numerous guest lectures at various institutions of national repute, corporates, as well as universities and colleges. He had undergone advanced training in the field of chromatography and spectroscopy at Thermo Fisher Scientific UK and Italy, and also at Nadu Corporation, Japan, China, and Singapore. Currently, sir is working with Toshbin Analytical Private Limited, Mumbai as a product consultant. With this brief introduction, I request the coordinators to welcome Girijan sir with e-bouquet. Now I request Girijan sir to start his session entitled Basics and Applications of UV Visible Spectroscopy. So over to Girijan sir.
Yes, Girijan sir.
So a very warm welcome to all the participants. A very good afternoon to all of you. And I had a privilege of being with you for this session. I mean, this is my second session with you people because I had already completed one session on the 1st of March on GCMS. So today I have a different topic to talk about UV visible spectroscopy, basic principles, and some of its important applications. And all of you will agree with me, UV visible spectroscopy is one of the most widely used analytical techniques across the segments. Not only in industry, but also in academia, UV visible spectroscopy is one of the most widely used analytical techniques. It's a very, very simple technique, but yet it's a very powerful technique. So you can do a lot of different types of analysis using UV visible spectrophotometer in different segments like pharmaceutical industry, food industry. You name an industry, we have got a lot of applications in UV visible spectroscopy for that. So I will start sharing the screen because today I have limited time, 90 minutes. I hope all of you can see the slide.
Yes,
hello.
sir. Yes, sir. Yes, sir.
Slide is visible. Okay, good.
Yes, sir.
So the topic is basics of UV visible spectroscopy, instrumentation and applications. I'll be very briefly discussing about the UV visible spectrophotometry instrumentation also, but that's also very, very important for us to understand, and some of the basic applications. And I know you had already listened to different spectroscopic techniques during this FDP. I mean, I think you had a lecture on FTIR also, I think 2 days back. And they're all basically, when you talk about a spectroscopy technique, they're collectively called identification techniques. And we have different types available today. Starting with the basic one, UV visible spectrum, it is always topping the list. And the simplest one also, it's the simplest technique. UV visible infrared, today it is FTIR. We have Raman, we have atomic absorption, inductively coupled plasma, AES, and when it is connected to MS, it becomes ICP-MS. We have fluorescence, we have nuclear magnetic resonance, we have mass spectrometry, and there are many others also. So these are some of the important analytical techniques that are widely used both in industry and academia. So spectroscopy technique, when we talk about spectroscopy, it can be broadly divided into two types: atomic spectroscopy and molecular spectroscopy. The division is based on what are the changes taking place when a spectroscopic technique is being carried out, whether the changes are taking place at the atomic level or whether the changes are taking place at the molecular level. See, in spectroscopy, what we normally do, what we normally study, when any sample or matter, I can call sample as matter, when any sample is subjected to a certain type of radiation, some changes are going to take place. And if these changes are taking place at the atomic level, then we have atomic spectroscopy techniques where we have atomic absorption spectrophotometry, inductively coupled plasma. They're all coming under that heading. But if the changes are taking place at the molecular level, we have got molecular spectroscopy, NMR is there, Raman is there, UV visible is there, FTIR is there, and the fluorescence is there. So different types are coming under molecular spectroscopy also. So today we are going to briefly discuss about UV visible spectroscopy. And the moment you talk about spectroscopy techniques, the first thing that is coming to our mind is the electromagnetic spectrum. And all of us know we have different types of radiations available today. And different types of radiations are characterized by two basic properties. One is the energy content of the radiation, and second one is the wavelength range of the radiation. And they are different. From radiation to radiation, the energy content is going to be different, and the wavelength range is also going to be different. So we have plenty of them. And if we try to arrange all these radiations in a certain sequential manner, an orderly manner, we get what is called electromagnetic spectrum. And what I have shown on the slide is a part of electromagnetic spectrum. And they are all arranged in a proper manner, in an orderly manner. How? They are all arranged. Suppose if you are looking at the slide, and if you look from the left-hand side to the right-hand side, you come from the left-hand side to the right-hand side of the slide, you will see the energy of the radiation is decreasing. That means on the extreme left-hand side, you can see some very, very powerful radiations like gamma radiations, cosmic radiations, X radiations. That is normal, our X-ray. They're all highly energetic radiations, very, very high energy content radiations. But when you go to the extreme right-hand side, we have radiations like radio waves, microwaves, terahertz region, and they are all very low energy radiations. And we all know energy and wavelength are inversely proportional. That means when the energy is more, your wavelength is going to be very less. And if the energy is going to be less, the wavelength is going to be more. I mean, it's an inverse relation. So on the extreme left-hand side, we have got very, very powerful high energy radiations. That means low wavelength radiations. And on the extreme right-hand side, we have radiations which are very low in energy content, but their wavelengths are going to be very wide. And our UV radiation and visible radiation, infrared radiation, they are all in the middle part of the spectrum. So when you, as you see on the slide, UV is coming on the left-hand side of visible. That means UV is more powerful than visible. Visible light is our sunlight. And visible light is more powerful than infrared radiation, infrared light. So infrared light has got lower energy content compared to UV radiation. And one more thing we know, UV radiation is a destructive radiation. That is why we tell people when a UV lamp is on, don't continuously look at the lamp with a naked eye. You need to have a eye protection. Otherwise, these radiations has the capability to kill the cells in your eyes. And there are cases where people have gone blind, continuous exposure to UV radiation. UV radiation is a destructive radiation. IR radiation is a non-destructive radiation. It's only heat radiation. So UV, and when we normally in a spectrophotometer, we call it as UV visible spectrophotometer because the spectrophotometer works both in the UV range and in the visible range. So when we consider a UV visible spectrophotometer, it can cover UV range, and all of us know the practical recording range in a UV spectrophotometer. Suppose tomorrow you are getting a pharmaceutical sample and you need to record a UV spectrum of the sample using a UV spectrophotometer. Normally we record it from 190 nanometer to 360 nanometer. That's a recording range of UV, 190 nanometer to 360 nanometer. And then from 400 nanometer to 800 nanometer is the visible range, where we normally carry out colorimetric estimations, colorimetric analysis. So people may ask, many times students have asked me, if UV is up to 360 and visible is starting from 400. So there is a gap from 360 to 400, what happens? And this region, 360 nanometer to 400 nanometer is called the overlapping region. Because in a UV visible spectrophotometer, you have two different lamps. One lamp is meant for UV region, another lamp is meant for visible region. UV region, all of us know we use a D2 lamp, deuterium lamp. which has got the maximum energy content in the UV region. So D2 is meant for UV, whereas in the visible region, we are going to use a tungsten halogen lamp. So tungsten halogen lamp is there, D2 lamp is also there. And if you are working on UV and visible, suppose you are recording the spectrum both in the visible range and UV range continuously, in the, from 400 between 400 and 360 nanometer, your lamp will change over. Automatically, the lamp will change over. When the scanning enters the UV region, your tungsten lamp will be automatically put off, and D2 lamp will be automatically, it will become on. So it is the lamp changeover region, and that is why it is called overlapping region. The exact wavelength at which the overlapping has to take place can be set by the service person. So 360 to 190 is the normal recording range for a UV spectrophoton. And you know, all these radiations can propagate through a medium in the form of a transverse wave. The transverse wave, when you look at the sea, how the water waves are coming, they're going up and coming down, going up and coming down, and it is getting continuously repeated. So there are maximum points and minimum points, what we normally call as the crest points and the trough points. So it is a transverse motion, and when it is propagating through a medium in the form of a transverse wave motion, it has got certain fundamental properties, like it can undergo transmission. The radiation can undergo transmission, it can undergo absorption, it can undergo refraction, and it can also undergo reflection. So all these are basic properties of a radiation when it is propagating through a medium. Now, what happens when light strikes a molecule? This is all what we are going to study using a specific spectroscopic technique. When light means any form of energy, when it is coming and hitting a molecule, our sample molecule, what actually happens? So there are two types of possibilities. Maybe the incoming light has got very low energy, or the incoming light has got high energy. So only two types. Either it is a low energy radiation that is coming and falling on the molecule, or it is a high energy radiation that is coming and falling on the molecule. In either way, the changes are going to be different. Changes are not going to be same, they are going to be different. So if a high energy radiation is coming and falling on a molecule, electronic transitions are going to take place. And what we are going to see what is that. And when a low energy radiation is coming and falling on the molecule, there are two types of changes that can occur. One is a vibrational transition. I think you had already listened to an FTIR lecture. So in IR, we are considering only molecular vibrations. We are concerned with only vibrational motions that are taking place in a molecule when it is subjected to IR radiation. There is one more possibility when a low energy radiation is coming and falling on the molecule. Rotational, the molecule can rotate about its axis, normally studied by microwave spectroscopy, or it can also be studied by Raman. So vibrational transitions or rotational transitions are caused by low energy radiation, whereas electronic transition is caused by a high energy radiation. So what is happening in the case of UV? UV is always a high energy radiation. So only electronic transitions will take place in the case of UV. So these are the different possibilities. If you look at the slide, you can see these are the different possibilities. See, when you consider a sample, when you consider a molecule, when you consider a sample at normal conditions of temperature and pressure, it has got a certain amount of energy associated with that. And this is what is known as the ground level energy or the ground state energy, always denoted by E0. So that is the notation we use. E0 is the amount of energy that is associated with any sample, with any molecule at normal conditions of temperature and pressure. Now you imagine some form of radiation is coming and falling on the sample. Now, depending upon whether the incoming radiation is a low energy one or a high energy one, now you know different possibilities are, three different possibilities are there. Okay. In the case, suppose if there is a very, very low energy radiation, like a microwave radiation is coming and falling on the sample, what will happen? Rotational transitions are taking place. And if you consider a molecule as a whole, there are different energy levels. The basic energy level is E0, and there is a upper level like E1, then E1, E2 like that. There are different energy levels. So the E0 is the base level, and E1, E2, they are all called excited levels of energy state. Now between one E0 and E1, so the E0 is one electronic level and E1 is an excited electronic level. Between E0 and E1, there are different vibrational levels, like V0, V1, V2, like that, different vibrational levels. So if higher radiation is coming and falling on the sample, there will be transition from one vibrational level to another vibrational level. So for example, it can go from V0 to V1, V1 to V2 like that. And all these are happening within the same electronic level. It is between V0, E0 and E1. So vibrational levels, energy has come down. Now, if you consider microwave is coming and falling on the sample, within the same vibrational energy levels, like between V0 and V1, there are different rotational levels. Maybe you can call it as R0, R1, R2 like that. So if a very low energy radiation, like a microwave radiation is coming and falling on the sample, there will be transitions between these rotational levels from R0 to R1 or R0 to R1 to R2 like that. It can happen. That is the lowest energy transitions that can take place in a molecule. Microwave, very, very low energy, rotational transitions can take place. Then infrared, vibrational transitions can take place. There is slightly more energy than rotational one. And finally, the electronic transitions. Here it is caused by your UV radiation, because UV radiation is a very, very powerful radiation, and UV radiation will cause an electronic transition. And that is the most powerful transition that is taking place in a sample molecule. Now, what is electronic transition? Again, you know, as a molecule at the normal standard conditions, temperature and pressure condition, normal conditions, it has got a certain amount of energy associated with the molecule. We call it as ground state energy, E0. Okay. And then assume that UV radiation is coming and falling on this molecule. Now, depending upon the type of molecule, that's a very important statement. Depending upon the type of molecule, the molecule will absorb some quantity of energy from the incoming radiation, and then the total energy will go to a higher level. That means from E0, it will go to E1. E1 is called an excited state of energy, and E0 is called the ground state of energy. Okay, now the difference between these two, delta E is equal to E1 minus E0. That is equal to, we can write it as h nu. That is the quantum of energy taken by the sample when it was exposed to UV radiation. Now I highlighted depending upon the type of sample. What does it mean? So when you are exposing all the samples to UV, my question is whether all the samples can absorb UV. Normally I ask this question to students, but so I get a lot of mixed answers. The answer is not all samples can absorb UV. Some samples will absorb, majority of the samples, especially luckily in our pharmaceutical industry, majority of the pharmaceutical compounds will absorb UV, but there are some compounds which will not absorb UV. So even if UV radiation is coming and falling on the sample, the radiation will simply pass through. It will not get absorbed. And those type of samples are called UV transparent samples. I can give an example, carbohydrates. Carbohydrate study is a very, very important application in food chemistry, and we do it by HPLC. And in HPLC, a very prominent detector, you know, you have already listened to an HC lecture. Variable wavelength UV visible spectrophotometer is a very, very popular detector for normal HPLC. But we don't use a UV detector for detection of carbohydrates or sugars. We use a RA detector, refractive index detector, because carbohydrates will not absorb UV. UV transparent sample. Similarly, saturated fatty acid methyl esters, FAME analysis. They will not absorb UV. and again a UV transparent sample. So there are other detectors which can be used for that. Okay. Now, so this is what is basically in electronic time. But now why we call it as an electronic transition? Now you know what is the, if you look at the structure of an atom, there is a central nucleus where all the protons and neutrons are concentrated. And around the nucleus, there are various orbitals, and these orbitals, the electrons are continuously orbiting the nucleus. So when the UV radiation is coming and falling on the sample, an electron from the outermost orbital gets energized, absorbs energy, and it is pushed to a higher level. So that means one electron is getting elevated from a lower energy level to a higher energy level. Okay. The electron is getting elevated, electron is getting transitioned from a lower level to higher level. We call it as an electronic transition. And to elevate an electron from a lower level to higher level, the incoming radiation has to be very powerful. UV has the required power. IR doesn't have. That is why IR cannot induce an electronic transition. It can induce only vibrational transitions in the molecule. So electronic transition is all that is what is taking place in the case of UV radiation. Absorbing, so suppose my molecule is M, and M I am going to expose it to a certain radiation H nu. That's energy. There are two different possibilities, two different scenarios. So whether my molecule can absorb certain quantity of energy H nu, and it can go to an excited state. M is getting converted into M star. M is the ground state, M star is the excited state. But this excited state cannot remain there for a long time. Its shelf life is extremely low. Immediately it will try to come back to the ground state. M star will come back to M, and in that process, it will give out the energy that is consumed in different ways. It can come out in the form of fluorescence. That is what we study in fluorescence spectroscopy. It can come out in the form of light. It can come out in the form of phosphorescence. So these are the possibilities. But anyway, the whatever energy is absorbed by the molecule will come out. This is one possibility. Second possibility, when the molecule absorbs energy and it goes to an excited state, M is going to M star. Instead of coming back to the starting material, that means instead of coming back to M, this M star will be converted to a totally different entity. M star is getting converted into N. N is a different entity, and this is nothing but a photochemical reaction. So that means a totally different entity is formed. So two possibilities. Now, when you look at the atomic structure, the energy levels, these are basically what we have studied in quantum mechanics. The electronic transitions can take place across different energy levels. And what are the energy molecular orbitals? We have got sigma orbital, we have got pi orbital, we have got n orbital, and then we have got pi star and sigma star orbitals. Now, sigma and pi are always, and n are always occupied levels. Sigma, pi, and n orbitals are always occupied with electrons, whereas pi star and sigma star are normally unoccupied levels. So there is a freedom for the electrons from an occupied level to go to an unoccupied level. So that means the transitions can take place from an occupied level to an unoccupied level. And that is actually what is happening in the case of UV spectrophotometer, UV spectroscopy. So what about transitions can take place? One electron can go from sigma to sigma star level. It was originally at sigma level. It can get transitioned into sigma star. But this is a very, very high energy transition. This transition, sigma to sigma star transition, involves the maximum energy. And this is displayed by, or it is happening in the case of only saturated hydrocarbons, alkanes. Saturated hydrocarbons, alkanes. And this can happen somewhere around 140 nanometer to 150 nanometer. So normally when I ask a question to students, suppose you are using normal hexane as a solvent in UV. Normal hexane is a very, very widely used solvent in UV for making solutions. And people say normal hexane is a UV transparent sample. So I ask a question to students, whether do you really agree with this comment? Normal hexane is a UV transparent sample. And most of them say yes. I mean, it's a UV transparent sample. It does not absorb UV radiation. I said, I normally tell them the concept is different. In the when you consider our UV spectrophotometer, which we are using, where the range of the UV visible spectrophotometer, most of the UV visible spectrometers available in the market, they are starting from 190 nanometer to say around 1000 or 1100 nanometer. So starting from 190 nanometer. So under the conditions of the UV spectrophotometer, certain whatever transitions are taking place below 190 nanometer, we are not able to see. The instrument is not able to see. So that means we feel the compound is UV transparent. So that means I can slightly modify the statement. The compound, say for example, normal hexane is a UV transparent sample under the conditions of our equipment, because it is absorbing. Sigma to sigma star transition is taking place even in the case of a saturated hydrocarbon like normal hexane, but it is happening somewhere around 140 to 150 nanometer range, which our instrument cannot read, because our instrument is starting from 190 upwards. So that is the reason. Okay, so sigma star transitions are possible for very high energy involvement, saturated hydrocarbons, alkanes. Carbonyl compounds, most of the carbonyl transitions we can note and we can see and we can record. And that is basically sigma to pi star transition, carbonyl compounds. Unsaturated compounds, pi to pi star transitions, very easy for us. Very easy, we can record all those, we can study all those, we can measure all those transitions, pi star transitions. Then N sigma star transitions for samples containing halogen atoms, oxygen, nitrogen, sulfur, or N pi star. Again, certain carbonyl compounds can exhibit an N pi star transitions also. So these are, just again, whatever I told you, the same thing. See, sigma to sigma star, then pi to pi star, N to sigma star, and N sigma star, N sigma star, N pi star, and pi star. Different types of compounds will undergo these type of transitions. And if these transitions are falling in the range of our equipment, that means from 190 nanometer upwards, we can easily measure all those. But many of the pharmaceutical compounds, they all fall within the range. So in the range of 190 nanometer and upwards. And that is the reason why we are able to use UV very, very effectively in pharmaceutical industry. Okay, now what is the reason why what type of samples can absorb UV? Some samples containing some single bonds, not all single bond, some type of single bonds. Double bond, any sample containing double bonds unsaturation will definitely absorb UV. Conjugated double bonds, if these double bonds are in conjugated position like single double position, then they are highly UV absorbing compounds. Simple example is benzene solvent, 6-membered conjugated aromatic system, highly UV absorbing. 254 nanometer is the lambda max, approximately around 254 nanometer. Some metal and inorganic compounds, nitrogen, oxygen, sulfur, halogen containing compounds. So these are the types of samples which can absorb UV radiation. So the basic requirement for a sample to absorb UV radiation is the sample molecule has to contain one chromophore. That is the basic requirement. If the small chromophore is not there, then the absorption will not take place. And what is a chromophore? Molecules having unsaturated bonds or free non-bonding electrons that can absorb radiation of relatively low energy are called chromophores. We are stressing the word relatively low energy. That means if it goes to high energy, that means it will go below 190 nanometer. We will not be able to see, we will not be able to measure. So when it is coming to 190 and upwards, the energy is coming down. And when wavelength is more, your energy is less. Inverse relation, we know that. So a molecule or any molecule having unsaturated bonds or free non-bonding electrons that can absorb radiation of relatively low energy are called chromophores. It's nothing but it can be considered as a functional group. Examples include alkenes, again unsaturation, alkynes, triple bond systems, ketones, carbonyl compounds, aldehydes, CHO functional group, phenyl compounds, there's a possibility of benzene ring, and all other aromatic species. So a chromophore refers to a functional group that absorbs light. For example, carbon-carbon double bond, carbon-oxygen double bond, nitrogen-nitrogen double bond, nitrogen-oxygen double bond, like that. Okay. And if any of the symbol chromophore is conjugated, that means a molecule can have more than one chromophore. That possibility is always there. The requirement is at least one chromophore should be present, but there are molecules which contain multiple chromophores and they are known as multiple chromophoric systems. And there is one more concept called oxochrome. And what is an oxochrome? It is again a functional group, but this functional group by itself, it will not absorb UV. It has no capability to absorb UV radiation, but it has the capability to influence the absorption due to chromophore. That means suppose you imagine you have got a molecule, it contains a chromophore, and if there is also an oxochrome, this oxochrome will influence the absorption because of the chromophore. In what way it is going to influence? Your absorption will go to longer and longer wavelength if a chromophore, if an oxochrome is present. Suppose because of the chromophore, suppose your sample is absorbing at, say, for example, 250 nanometer. And if an oxochrome is also present in the molecule, then probably your absorption will go from 250 to 270 or 290 or 300. It will go to longer wavelength, number one. And number two, it can also increase the intensity of absorption. Again, suppose your molecule contains a chromophore, and because of the chromophore, your sample is absorbing UV, and what absorption you are getting? Say 0.5. You are getting 0.5 absorption. That's the intensity of absorption. Now, if an oxochrome is present, this intensity can increase, say from 0.5, it can go to 0.6, from 0.7 or 0.8. So it is acting in two ways. One is increasing the intensity of absorption, and it is also increasing, changing the absorption, the wavelength, shifting it to long. The lambda max will be shifted to longer and longer wavelengths. So these are the two influences an oxochrome will have on the working of a chromophore. And few examples of oxochromes: hydroxyl groups, OH, OR, NH2, amino groups, SH, thiol groups, halogens, they all can act as oxochromes. And when your sample molecule contains more than one chromophore, what is our inference? Your lambda max will be shifted to longer and longer wavelength. For example, I am just shown an example here. I got a diolefin, a molecule containing two double bonds, but they are not in conjugated position. They are unconjugated. And their lambda max is somewhere around 190 nanometer. Now, if it goes to conjugated, the same diolefin, if the double bonds are in conjugated position, lambda max will go to 270. From 190, it will go to 270. And there is another example, triolefin, that is a molecule containing three double bonds. You can see the lambda max is already gone to 250 nanometer. So each double bond is a chromophore. So that is why in the case of UV, suppose if your molecule contains unsaturation, it is an excellent member for UV absorption. You can easily measure the absorption in the UV spectrophotometer. The solvent cutoff value, especially for when you are working with a UV spectrophotometer, you have to be very careful because you have to make a very, very dilute solution of the sample, because the detector is so powerful that a very, very dilute solution only can be used. Otherwise, detector saturation will take place. It will go to non-linear part of the detector. That means your quantitation result will be wrong. So you have to prepare a very, very dilute solution, and you have to see that your absorption readings lie in the linear part of the detector. Normally, we advise the customers to make the solution or adjust the dilution in such a way that you get an absorption reading between 0.3 and 0.8. Because we normally have a absorption range on the y-axis. UV spectrum is again a UV XY graph. X-axis is nothing but the wavelength in nanometer, and y-axis is nothing but the absorption value. So normally we keep a range of 0 to 1. 0 to 1 is the absorption normal range we keep. So in this 0 to 1, your absorption reading should be between 0.3 to 0.8. If it is more than 0.8, that means the concentration of the sample is more. You have to dilute it slightly more. And if the absorption is less than 0.3, that means you made it too dilute. You have to slightly add the sample. So this is the concept. Okay. Now, when you are using a solvent for making a dilution, there are different solvents are available, and you should know what is the solvent cutoff value. So I've shown so many different solvents. For example, you take chloroform. Chloroform is 240 nanometers the cutoff value. Chloroform is an excellent solvent for many other pharmaceutical drugs. So what is meant by cutoff value? So it means chloroform has got a value of 240 nanometer. That means above 240 nanometer, the absorption of chloroform will drastically decrease, but below 240 nanometer, absorption of chloroform will drastically increase. So that means tomorrow you are getting a drug to be analyzed by UV, and this drug is highly soluble in chloroform, and you are going to make a solution in chloroform. And now you imagine your lambda max of your drug is 220 nanometer. Can you use chloroform? You cannot, because when you are measuring at 220 nanometer, your chloroform is also going to absorb heavily. So you'll get an excessively high absorbance value. So you cannot use chloroform. You'll have to look for a different solvent. But at the same time, if this drug has got, say, lambda max 310 nanometer, you can use chloroform without any problem because it is much above 240. So at that 310 nanometer level, chloroform will be practically transparent. You can use it. So you should know the solvent cutoff value for each solvent before you use it for diluting the sample for measurement in the UV system. This you can get from the literature, or this you can get from the solvent manufacturer also. Now this is a spectrum of anthracene. Anthracene, it's a polyaromatic hydrocarbon. 3 benzene rings are fused together, and each benzene ring, we know it's a chromophore. So that means there are 3 chromophores are there in a molecule of anthracene. And if you look at the UV spectrum of anthracene, we can see the lambda max is going more and more towards 400 value. That means it is already gone to 360. I mean, it has gone to very, very long wavelength, and further it can even go to your visible range. The moment it crosses 400, it is going to visible range. So it has gone to very long wavelength because of the presence of 3 chromophores in the molecule. Another example is vitamin A. Vitamin A is an oil-soluble vitamin, and it is an excellent example for a conjugated system. You can see the conjugation in the structure of vitamin A, and because of this, it is a highly UV absorbing material. Normally, vitamin A we are analyzing by HPLC with a UV detector. It has got excellent UV absorption characteristics. So this is it's a highly conjugated system. And when you talk about conjugation, it will become an excellent UV absorption characteristics. Now there are 2. Today we are using UV as a quantitative technique. Most of the time we don't use today nowadays. You don't use a UV as a qualitative. If you talk about FTIR, FTIR is an excellent qualitative analytical tool. You can find out what are the functional groups present in the molecule looking at the IR spectrum. But it is difficult for us to understand those information by looking at a UV spectrum, because many different samples will give you very, very identical UV spectra. That's a major limitation for UV, and that is the reason why we are not using UV nowadays as a qualitative analytical technique. At the same time, it's an excellent quantitation technique. And when you're going to do a quantitative analysis by UV, there are 2 important laws. And the third one is the combination of the first 2. And what is the first 2? Lambert's law absorbance is directly proportional to the path length of the absorbing medium. We are going to make a solution, and all of us know, all of us must have, all of you must have done measurements in UV. You prepare a dilute solution and you take in the cuvette. Normally we use a quartz cuvette. If the measurement is to be done in UV region, we have to use a quartz cuvette. Glass cuvette cannot be used because glass absorbs UV radiation. Glass cuvette can be used in visible, but not in UV. UV, we have to use a quartz cuvette. And the standard cuvette is always 1 centimeter by 1 centimeter square cell. So the path length is 1 centimeter. Path length means the distance through which the light is passing through in the solvent, in the solution. That's the path length. When I use a 1 centimeter by 1 centimeter cell, my path length is always 1 centimeter. So absorbance, whatever reading I am going to get when I keep the sample in the sample compartment, whatever reading I am going to get on the instrument, that is my absorbance value. And this absorbance is directly proportional to the path length. This simply means if I increase the path length, my absorbance also will increase. Direct proportion. Lambert's law. Second one is Beer's law. Beer's law states absorbance is directly proportional to concentration. Concentration of your target compound. That means if the concentration is going to be more, your absorbance value is also going to be more. Again, it's a direct relation. Now, if you combine these two, Beer's law plus Lambert's law, you get Beer-Lambert's law. Absorbance, capital A, is directly proportional to concentration as well as the path length of the medium. This is what is Beer. Most important law in a spectrophotometric estimation. Whether you are going to do the estimation by UV, FTIR, atomic absorption, ICP, fluorescence, if you are going to do a quantitative analysis, Beer-Lambert's law must be obeyed. Otherwise, your quantitation result will not be proper. So this is the equation. Capital A is the absorbance value is equal to epsilon multiplied by L multiplied by C. What is epsilon? It's a constant. It's nothing but the molar absorption coefficient of the compound at a given wavelength. That is what is known as molar absorption coefficient. And what is molar absorption coefficient? It is the absorbance value of a 1 molar solution in a 1 centimeter cell. Absorbance value of a 1 molar solution in a 1 centimeter cell is your molar absorption coefficient. It is a constant for a given compound at a certain wavelength. It's a constant, epsilon. Then L. L is again path length. That is also a constant because you are going to use the same cuvette cell for your standard preparation as well as sample preparation. You are not going to change the cell. So L is always a constant. So if I take out these two constants, I remove epsilon, I remove L, then this equation becomes A is directly proportional to C. That means absorbance is directly proportional to concentration. You can call it as Beer-Lambert's law. And this is to be obeyed whenever you go for a quantitation by any form of spectroscopy. And if you get an absorbance value, you can convert it to transmittance value also. Very simple equation, a very, very basic thing. Absorbance is equal to 2 minus logarithm to the base 10 of percentage transmittance. So if I know what is the percentage transmittance, I can convert it into absorbance value. Or if I know what is absorbance value, I can convert it into my transmittance value, both ways. So A is equal to 2 minus logarithm to the base 10 of percentage transmittance. Now, what is the effect of analyte constant? This is a very critical thing when you are preparing the solution. Your sample has to be very, very dilute solution. If the sample concentration is more, your absorbance reading will go to the non-linear part of the non-linear range of the detector. Any electronic detector has got a linear range and a non-linear range. Whether you talk about FID in GC, or whether you talk about a detector in HPLC, whether you talk about a UV detector, any electronic detector has got a linear range. So linear range means when you are increasing the concentration, so for example, so UV, when you are increasing the concentration, the absorbance value also should proportionately increase. That is called the linearity. This can happen up to a certain point, up to a certain level. Beyond that, even if you increase the concentration, the absorbance reading will not increase. It will become a plateau. And that is what you can see in the slide. The red dotted line, that's a plateau. And that is the nonlinear range. So your absorbance readings goes to that range, then your quantitation is going to be totally wrong. So what is required is you should be getting the absorbance reading in the linear range. Linear range, you can see, I will say I have prepared a 5-point calibration here. You can see 5 different concentrations we prepared, and all the absorbance value are falling in a straight line, and that is the linear range. That is for the working range of my equipment. So your measurement should be in the linear range. Straylight, you must have heard about Straylight. Even USP also has got Straylight specifications. Now, you are all pharmaceutical people, if you look at method number 857, that is the latest USP method 857 is the validation of UV visible spectrophotometer. It's a long chapter and little complex to understand. And Straylight, so there are so many parameters are given in that. So one is Straylight. Straylight is, in very simple definition, Straylight is any unwanted radiation, any unwanted wavelength, any unwanted light that is reaching the detector is called Straylight. So for example, a very simple example, suppose I am going to make a measurement of my sample in a UV spectrophotometer, and my lambda max is at 254 nanometer. So ideally, only 254 nanometer light should go to the detector. But unfortunately, along with 254, 260 nanometer is also going there, 250 nanometer is also going there. But these are all unwanted wavelengths. That means they are all collectively called Straylight. Any unwanted wavelength, any unwanted light that is coming to the detector is called Straylight. Straylight is not under our control. It is under the control of the manufacturer. It is basically a function of the hardware system. So when they are designing the instrument, they will try to reduce the Straylight as minimum as possible. You cannot design an equipment with 0 Straylight. That is not possible. But you can reduce the extent of Straylight in the system, in the equipment. So there are some high-end UVs. I will show you one or towards the last slide of my presentation. I will show you one or two equipments. But the Straylight specifications are very, very stringent. They say Straylight is 0.00001%, like that they say. But they will never write. Nobody will write Straylight is 0%, 0.0%. That nobody can write. We have to give some value. If Straylight is more, what will happen? Your sensitivity will reduce. There are 3 different ways of finding out the Straylight as per the USP recommendation at 3 different wavelengths, using 3 different, I think 3 or 4, 4 different regions using 4 different standards. At around 198 nanometer, we use 2% potassium chloride solution in water, and that is the standard we use. And similarly, there are 2-3 other standards also. You can actually refer to method 857 in USP. And some of the UV visible NIR spectroscopy applications, why UV has become so popular nowadays. For every sample, you can get a spectrum. Very easy sample concentration calculations, very user-friendly software. Calculations are very simple and affordable instrumentation. The cost of the equipment is also not prohibitive. So even education institutions, colleges, they can all afford to buy a UV. And UV is an excellent tool for the students also, maybe postgraduate students or PhD students, or even faculty members or having different projects. I mean, UV can be a wonderful equipment for all those type of work. So, and anybody can afford it also. It's not a very prohibitive cost. Then the limitations, as I told you, the problem is you cannot go for a qualitative identification because you get very little structural information, because different compounds will produce very similar spectra. That's the main problem we encounter when we go for a qualitative identification by UV. So that is why we don't do that now. We go for only quantitative estimations. But when you consider an FTIR, 90% of the people are using it for qualitative identification. Whereas FTIR also can be used for excellent quantitation. FTIR can be used for excellent quantitative analysis. But quantitation by FTIR is slightly difficult and complex as compared to quantitation by UV. UV is very simple, quantitation is very simple, nothing. But FTIR quantitation is a little complex. but it can be done. Now, what are the major areas? See, for example, agricultural applications, food industry. Food industry is a major segment where we can see the usage of UV. For example, in food, there are two important considerations as a consumer: color of the food product, consistency of the food product. Very, very important. It's appealing to your mind. So maybe today you go to a grocery store and buy a tomato ketchup from some XYZ company, and you know the color is very dark red, real tomato color, and the consistency, the material is very thick. It is not water. It is not very loose. It's very quite thick, and you are happy about it. And maybe after 6 months, you go to the same store and buy the same tomato ketchup by the same company, and this time you observe the color is little different. It has become a little pale yellow. And second thing, the material has become little loose. So as a normal consumer, so many different doubts will come to your mind. Whether the sample has undergone degradation, on storage, or any adulteration has taken place, all these will come to a normal consumer mind. And the manufacturers are also aware of this. So they want to keep the color, consistency, all those properties consistent from batch to batch. Okay, so what they do, we have got a color measurement software available with UV. So you can you can use a color measurement software of the UV color measurements. So variations in color can be determined, and then corrective steps can be taken. Similarly, we also add different types of additives in a food formulation. It can be an antioxidant, it can be an anticoagulant. I mean, these type of additives are added in extremely small concentrations. For example, benzoic acid, sodium nitrite. All these are there, but they are in very, very small concentration, but they are very critical. Their concentrations are very critical. And how we can use a UV for the estimation of quantifying those additives. Similarly, in beverage manufacturing, I mean, soft drink manufacturing companies, liquor manufacturing industry. I mean, there are so many different applications for color, consistency, or even additives. All these can be measured very consistently measured by UV spectrophotometry. Leaf analysis, chlorophyll, is a purely academic application. Chlorophyll, different types of chlorophylls are there. Chlorophyll A, chlorophyll B, like that. And depending upon that, the green color, the extent of green color also can change. Sometimes you can see some leaves are very dark green, some are pale green, pale yellowish green. These are all basically because of the difference in the chlorophyll content. And we can find out this by UV spectrophotometer in combination with the color measurement system. Biological applications, plenty of applications for UV, protein analysis, DNA, RNA quantification, kinetic studies like monitor rate of reaction or enzymatic reaction. These are all extremely critical in biotechnology or biological samples, serum manufacturing companies. So we have got different applications for UV, and we have got in the system, in the software, there's a feature. It's by default, there's a feature by kinetic mode. So using this kinetic mode, you can also go for kinetic monitoring of certain reactions, and you can evaluate some enzymes. I mean, all those are possible. So plenty of applications in biological field. Industrial applications, number one is pharmaceutical manufacturing. Metabolic pathways of drugs, kinetic studies, dissolution content, dissolution testing, and raw material analysis, intermediate testing in API manufacturing, content uniformity in tablets, cleaning validation in API manufacturing, nucleic acid analysis. There's a long list of applications in UV, in pharmaceutical industry for UV. Dissolution testing is one of the major applications. Dissolution testing, we use UV. Nowadays, even online UVs are also available. You can directly connect your dissolution tester to UV. And otherwise, we can take out an aliquot and measure and keep it back. So UV is an absolute must for this. You can even do a dissolution testing by LC also, but you consider what is the amount of time you are going to take. UV is much, much faster. So UV has become the preferred choice of analysis, especially for dissolution testing. Same way you can do it for content uniformity of tablets. Cleaning validation in API map, because all the pharmaceutical drugs are batch processing methods. So when you change over from one batch to another, your the process equipments have to be cleaned, and there's a cleaning validation procedure. Either you have to take a swab or you have to take a rinse and then take it and then test it for any residues on the previous batch or previous drug. We can do it by 3 different methods. You can do it by HPLC, you can do it by total organic carbon TOC, or you can do it by UV. And UV is the fastest. So many people do it by UV because it gives you very, very fast result. HPLC also can be done, but then the time, the procedure, all those. So UV is much faster. Similarly, in the field of chemical manufacturing also, quality control, raw material analysis, kinetic studies, intermediate analysis. So plenty of applications in pharmaceutical manufacturing as well as chemical manufacturing. Academic and government applications, environmental testing, contaminants in streams and water sources. I will show you one example after this. How UV can be used for contaminant analysis in water. And that's a very major application in power generation companies, like say, for example, Tata Power, or major petrochemical companies or fertilizer companies. And research laboratories, CSIR labs and universities, optical materials. Nowadays, people are working on different types of optical materials, thin films. And there we are more interested in the reflective properties and the transmission properties of all this. We have got different attachments for doing all this. So purely research applications, especially in academia. And then for students also, it's a wonderful equipment for the projector. So UV is one instrument used across industry as well as academia. And UV spectroscopy, what are the strengths if you look at? An easy to use method, it's a very robust method, and it offers you very good precision for making quantitative measurements of drugs. Not only drugs, I am showing it, drugs and so many other compounds. Routine method for determining some of the physicochemical properties of drugs, which need to be known before you go for a formulation. Some of the problems of the basic method can be solved by the use of derivative spectra also. There is a totally different chapter. Time will not permit me to go into it. I'll just tell you what is derivative spectroscopy in one of the slides. Limitations, only moderately selective. That is what I told you, because you cannot differentiate one compound or one molecule from another molecule by looking at the UV spectrum. Both will give almost similar type. That's a major limitation. Okay. UV spectroscopy in pharmaceutical analysis, applications in pharmaceutical area, a robust workhorse method for the quantification of drugs in formulations where there is no interference from excipients. If excipients are not going to interfere, you can even go for a quantitation in a formulation. Well, that is something like multi-component analysis. Used to determine the release of drugs from formulation, that is dissolution testing. And I also told you about content uniformity testing. Can be used to monitor the reaction kinetics of drug degradation and assay of pharmaceutical raw materials as per pharmacopoeial methods. See, this is an example of one is hydrocortisone and one is beta-methasone, 2 different steroids. If you go for a IR analysis of this, you go for a FTR analysis, you can easily differentiate between these two because the IR spectra will be different. But if you go for UV analysis, I have shown the UV spectrum of both hydrocortisone and beta-methasone on the left-hand side. You can say they are more or less identical. Almost you can say like overlapping. The lambda max is also very close by. And you will not be able to differentiate hydrocortisone from beta-methasone by looking at the UV spectra because they are more or less identical. So that's the main disadvantage. I should not use the word disadvantage, the major limitation of UV. At the same time, if I want to quantify this steroid, then I can use UV. So that is why UV has become an excellent quantitation technique rather than a qualitative technique. Just one slide on derivative spectroscopy. Derivative spectroscopy, what we are going to do is we are going to plot the rate of change of absorbance against the wavelength. That means we are going to plot delta A by delta lambda. Delta A by delta lambda is the rate of change of absorbance with wavelength against wavelengths. So that is what we are going to plot. Okay. And what is the advantage of a derivative spectroscopy? Derivative spectrum contains sharper peaks where better location of peaks and wavelength maxima can be achieved. Suppose when you are recording a spectrum of a compound, suppose you imagine there is maybe an impurity or a different compound in your sample, but they are not properly separated. Both the peaks are not well separated. And you are getting one main peak, and on one side you are getting a small hump. That means they are not properly separated. In those type of scenarios, if you apply a derivative spectroscopy technique, you can separate it, and then you can identify the lambda max of both, and then you can quantify them separately. And there are four different derivative spectrum order. There are the four orders are there. Derivative orders are there, 1 to 4. But as you increase the order of derivatization, what you have to keep in mind, your noise level also will increase. When noise level increases, that can actually reduce your signal to noise ratio. Okay. So as the derivatization order increases, like first order, second order, third order, fourth order, up to 4 orders are possible. So when you're going from first order to second, second to third, third to fourth, that means you're increased the order of derivatization. There is a possibility that your noise level also will increase. And when the noise increases, sensitivity will be down. So if you're okay with your zero order spectrum means the spectrum you are getting when you are keeping the sample inside the equipment, whatever spectrum you are getting, that is called the zero order spectrum. And on this zero order, suppose if this zero order spectrum is okay with you, you are happy with that, it's acceptable, don't think of derivatization. Derivatization is carried out, derivative spectroscopy, very, very few people are using. I mean, it has got certain advantages, no doubt, but not very widely. So what you see at the top is called the zero order spectrum. And on the zero order spectrum, we can go for the first derivative, we can go for second derivative, we can go for third derivative, and finally we can go for the fourth derivative. So these are used in very, very rare cases where an overlapping is taking place, proper separation is not possible, and then you can separate it by using a derivative technique and then separately quantify. Okay, this is itself is a topic for discussion. So time will not permit me to go deep into the derivative spectroscopy. Now, final part, we are coming to the basic components of a UV spectrophotometer. You have a light source inside, and from the light source, the light is coming to the monochromator. From monochromator, it will come to a sample compartment. From sample compartment, it will come to the detector, and the detector is giving output. Then that is your PC and software. Then you will get an output also, your UV spectrum. Okay. Now, so this is what I have written: sources, the sources, and wavelength selectors. The monochromator. What is the role of monochromator? All of us know. Monochromator is nothing but the wavelength selector. Okay, so the wavelength, whatever the wavelength is required for the estimation, is selected by the monochromator. Because the light that is coming from the source is a polychromatic light. Polychromatic light means light having multiple wavelengths. That's called a polychromatic light. And monochromatic light means light having a single wavelength. So for our measurement, we require a single wavelength. So polychromatic light should be converted into monochromatic light, and that is what is the responsibility of the monochromator. Monochromator is converting a polychromatic light into monochromatic light. And then it will pass through sample compartment. That is a sample solution. And finally, it will come out of sample solution, go to the detector, and then you have your software and PC. And your instrument either can be a single beam instrument or a double beam instrument. That's also, I will show you one slide now. What is the difference between these two? So this is exactly what I told you. There is a source of light, and that light is emitting light. That source is giving out light, which is a polychromatic one. And that is coming and falling on the monochromator. Monochromator is called the wavelength selector. And this monochromator is converting the polychromatic light into monochromatic light. And this monochromatic light will be passing through the sample compartment. And finally, it will go to detector. What you see here in this slide is called a single beam spectrophotometer. Single beam means it has got a single path length. There is a single wave of light. In double beam, there will be two different light beams. I mean, we'll see how it is done. Okay, now what are the sources of light? And I told you in the case of a UV visible spectrophotometer, you have a UV range, you also have a visible range. Okay, and the UV range, that is your ultraviolet range, we are using a deuterium lamp, D2 lamp. And this D2 lamp you can use for 190 to 360 nanometer recording of the UV spectrum. And visible region, where we are going to do colorimetric analysis, we have got a tungsten halogen lamp. We can see it can operate between 320 nanometer or 350 nanometer, and it can go to very, very long length, 2500 nanometer, which is a NIR region, near infrared region. So we have two different lamps, and the lamp also can change over, as I told you, that is for the overlapping region between 360 nanometer and 400 nanometer. So wavelength selector. In the olden times, we were using filters. Filter was used as a monochromator, but now the time is gone. Filters have become what you call outdated. Now we are using a monochromator. And monochromator, what type of monochromator we are using? We are using a grating monochromator. And what's a grating monochromator? Grating monochromator is nothing but a glass plate. And it's a glass plate over which a thin film of aluminum is coated. It is done by vacuum coating. A thin uniform layer of aluminum vacuum coating is done. And then this surface, we are going to draw lines. We are going to cut lines on the surface. And what is the number of lines? In 1 millimeter, we can draw either 1200 lines, 1600 lines, 1800 lines, or even 2000 lines can be drawn in a distance of 1 millimeter. Not possible manually. In 1 millimeter, we cannot draw 2000 lines or 1800 lines. They're all cut by laser beam. We use laser for that, cutting. And more the number of lines, better will be the resolving power. You will get a much better resolution. If you consider 1200 line per mm grating and a 1600 mm per grating, 1600 mm per lines per mm grating will be much better. That will have a better resolution power. So more the number of lines, better will be the resolving power of the grating. And what we do, and this grating, so after cutting, this is called grooves, and this is mounted on a motorized turntable. We are keeping this grating, which is nothing but a glass plate with a uniform thin layer of aluminum, which is vacuum coated. And then we are drawn lines to extent of maybe up to from 1200 or even 2000 lines per mm. And this is mounted on a motorized turntable. It's a motorized action. The turntable can move, rotate. That means your grating also can rotate. Now you can see in the slide, polychromatic light is coming from the source. Maybe from the UV source or from the visible source, the polychromatic light is coming. It is coming and falling on the grating. And we have already given what is our wavelength required for the analysis, that we have already given the software. For example, what example shown here, for this particular estimation, we are given 612 nanometer is the required wavelength for a certain analysis. We want a wavelength of 612 nanometer to pass through the sample solution. This we have already given in the software. So accordingly, what will happen when you start the analysis, the turntable will rotate. That means your grating will rotate, and at one position, you can see only 612 nanometer wavelength will come out of the slit. There is a slit inside, in front of this. That means a small opening. Slit is nothing but a small opening. And through this opening, 612 nanometer wavelength light will come out. All other light wavelengths will be totally blocked. So only 612 will come out. And then that will pass to the sample compartment and then go to the. Suppose if you change the wavelength, instead of 612, suppose you say I want 290. 290 nanometer is my wavelength required, the grating will again rotate and go to a certain position when only 290 nanometer wavelength will come out of the slit. So that is how we select, that is how the monochromator, grating monochromator is selecting a monochromatic light from an incoming polychromatic light. Okay. Now this slit width is also very, very important. The slit means the opening. The opening is also very important. If the opening is very wide, what will happen? Different wavelengths will go together. That means your resolution will be extremely poor. Lower the, or you can say narrower the width, better will be the resolution. On the left hand side, you can see I have shown 5 different spectra. Spectra number 1, 2, 3, 4, and 5. And if you look at spectra number 5, where we used a 2 nanometer bandwidth opening, 2 nanometer, you can see you are getting a very, very broad peak. There is no resolution. You are not happy with the resolution. So what you do, from 2 nanometer, you reduced it to 1 nanometer. Spectrum number 4. When you reduced it to 1 nanometer, you can see 2 bands are separating, but still they are very broad. 2 bands are separating. Again, you are not happy. You reduce it to 0.5 nanometer. The spectrum number 3, 0.5 nanometer, your 2 bands have become little sharper. At the same time, you have another observation. Immediately after the first band, there is a hump, which indicates that there is one more entity in your sample, but it is not properly separated. So what you did, from 0.5, you came to 0.3. That is a spectrum number 2. Then you can see all the 3 are separating, all the 3 bands are separated. And to reduce the width or to increase the resolution, you again reduced it to 0.1 nanometer. Now you can see all the bands have become sharper and they are all separated. You can easily identify what are the lambda max for each one of them, and you can quantify them separately. This is the importance of your bandwidth choice. So nowadays, some of the latest research grade equipment, you have got variable bandwidth equipments are available, where the user can change what is the, depending upon the application, depending upon the requirement, the user also can change the bandwidth. And whenever you are going to do a quantification, and most of the time you will be using UV only for quantification, please note that the quantification is to be done, and the measurement is to be done at the lambda max value. Your compound may absorb at three different wavelengths, but at one particular wavelength, the absorption will be really maximum value. And that wavelength is called the lambda max value, lambda max. Every compound has got a lambda max. So if you are doing a quantification, you have to do the quantification at lambda max value. If you don't know what is the lambda max value, scan the sample first. Take a complete run. Record the spectrum from 190 nanometer to 360 nanometer and find out at which wavelength the absorption is going to maximum value. And that is your lambda max. And that lambda max value can be used for your subsequent quantitative analysis. Okay. Now, this also shows the importance of carrying out the analysis at lambda max value. I have prepared 5 different solutions of a drug, 5 different concentrations. Sample is same, 5 different concentrations. The first one is maximum concentration 10 to the power of -2 molar, then 10 to the power of -3 molar, 10 to the power of -4 molar, 5 into 10 to the power of -5 molar, and the lowest concentration is 10 to the power of -5 molar. So 5 different concentrations, right from a very low concentration to a high concentration. Some 10 to the power of -5 molar to 10 to the power of -2 molar. 5 different standard solutions were prepared. And they were all analyzed on UV, and the measurements were done at the lambda max value. You can see on the right hand side spectrum, you can see lambda max we have measured. And at this lambda max value, even we could measure the absorbance of the lowest concentration standard, 10 to the power of -5 molar. That also we could measure without any problem. Now you imagine we are going to do the same measurement at a different wavelength, lambda 1. I have shown it as lambda 1. Lambda 1 is a totally different wavelength. It is not lambda max, lambda 1. You can see I am not able to measure the absorbance of the lowest concentration 10 to the power of -5 molar, plus all other standard sample standard solution, the absorbance have drastically come down. So this is what is going to happen if you make the measurement at a different wavelength rather than making the measurement at lambda max value. So lambda max value measurement, that is the most important thing, critical thing, if you want a really good quantitative analysis. Okay. And sometimes we can, very, very rarely, we can make the measurement at a different wavelength. Maybe in case if there is an interfering compound in your sample, and that also absorbs the same lambda max value. In that case, to avoid the interference, you can try to use a different, you have to use your logical thinking, and you have to select a different wavelength where it may not be lambda max, it's a different wavelength, and the absorbance also will reduce, but still your interfering compound will not absorb. So you can selectively detect your compound. So this happens very, very rarely, not always. Okay. And coming to detector, of course, this electronics part, these are all, we have different detectors available in UV. EMT is the most powerful one, photomultiplier tube. Then we have got silicon photodiode. Then we have got indium gallium arsenide for NIR region. So different detectors are available. PMT is supposed to be the most powerful detector, and the PMT is also costly. So some of the high-end models we are putting PMT. And otherwise, we are using, and this is all the, I am not going to this, these are the basically electronic parts, photomultiplier tube, how does it function? And coming to general types of samples, three different, liquid samples and solid samples only can be analyzed by UV, not gas samples. Gas samples can be analyzed by FTR also. FTR can analyze all the three, gas, liquid, and solid. But UV can analyze only liquids and solids. Liquids can be of three different types, transparent liquid. Transparently, if you take distilled water, that's a transparent liquid. You take normal hexane, benzene solvent, they're all transparent liquid. You can directly keep the sample inside the instrument. You don't require any special attachment, and you can read the transmittance value directly. But if it's a turbid liquid, that's called translucent liquid, turbid, it's not very clear. Then you require a attachment which is known as integrating sphere, which we are providing, which we are offering that. It's called an integrating sphere. That is required in case if you want to measure a turbid liquid. And using integrating sphere, either you can measure the transmittance of the liquid or the reflectance of the liquid. And if the liquid is an opaque liquid means very highly colored liquids, highly black color, blue color, green color, red color. The color is so thick that the light cannot pass through. They are called opaque liquids. There you require an integrating sphere, and you can measure only the reflectance, not transmittance. So three different liquids. So we have got the attachments also for all those type of applications. Then solid samples. Solid samples, it can be a powder, it can be a coating, it can be a mirrored surface. So different types of solid materials are there, but you have to, you can measure only the reflectance of a solid surface. And for that, you require either an integrating sphere, or there is one more attachment known as specular reflectance attachment. So these two attachments are required if you want to study a solid sample. So all those attachments are available from our side. And when you talk about reflectance, there are three different types of, I am not going to details, specular reflectance, relative specular reflectance, absolute reflectance, diffuse reflectance. There are different types of reflectance, and we have got different accessories for measuring all those. Specular reflectance means very simple. It requires a very smooth and shining surface, and angle of incidence is equal to angle of reflectance. That is what is known as specular reflectance. When the light is coming on a very polished surface, it gets reflected from the surface. In this case, the angle of incidence I is equal to angle of reflection R. It's called as specular reflectance. And what is diffuse reflectance? You have got a powder sample. You have got a rough surface, uneven surface. So in that case, diffuse reflectance will take place. Diffuse reflectance means the light will come and fall on the surface. It will penetrate, and while coming out, it will come out in different directions. And that is what is known as diffuse reflectance. So if you want to study a diffuse reflectance, you require an attachment called integrating sphere. Without that, you cannot study a diffuse reflectance properly. Okay, chemical complexation. In some of the compounds, but the absorbance, the concentration of the sample may be so low that you will not get a very sensitive absorbance reading. So now by adding a certain reagent, this reagent will react with your compound and form a complex. It's called a complexation. And this complex will be having a very high absorbance. And from the absorbance value of the complex, you can actually find out what is the concentration of your original target. For example, this is a water analysis. I will tell you more on this. I have got only very few slides more now. See, I want to find out what is the concentration of nickel in water. At the same time, iron is also present in water. And both are having lambda max fairly close to each other. And the nickel concentration is very less. So iron can be a problem. So what to do? So in case if I want to very precisely quantify nickel, I will add a reagent, dimethyl glyoxime. Dimethyl glyoxime will react only with nickel. It will not react with iron. It only reacts with nickel and form a nickel dimethyl glyoxime complex. Right hand side, you see the complex formation. Then you see the absorbance value. Absorbance is very high. And from this absorbance, you can actually calculate what is the actual content of nickel in the sample. Okay, so analysis of absorbing samples. If the sample is going to be absorbing UV, absolutely no problem. You don't require an attachment. And this is what is actually being done in pharmaceutical industry. Most of pharmaceutical compounds, luckily for us, they're all absorbing compounds. So you don't require any special attachment. You can directly keep the sample, make a solution, take it in the cuvette and keep it in the sample compartment. Absolutely no problem. They are all, your samples are all absorbing samples. But if the sample is a non-absorbing sample, like a metallic components, I mean, applicable to water analysis, especially in water analysis, I mean, in huge petroleum companies, petrochemical companies, fertilizer companies, people are using water for steam generation, cooling tower. Steam generation, people are using very huge quantity of water for generating steam for their process applications. And if this water, this steam will be circulating to the plant and coming back, and during the course of circulation, they pick up lot of impurities, metallic as well as non-metallic. And these impurities get accumulated in water. And ultimately, when this water is again heated in the boiler, these impurities can form a layer inside the boiler. This is known as crust formation. The crust formation is a very dangerous thing because too much of crust formation is there. When the boiler is heated, there can be an explosion of the boiler. It's a very serious safety hazard. Boiler can explode. It's a major problem. So they want to continuously monitor what are the concentrations of metallic and non-metallic impurities in water. So for example, magnesium, iron, aluminum, calcium, whatever it is. But then the moment you say metallic impurities in water, what is the instrument that is coming to your mind? Number one, atomic absorption or ICP. But both are quite expensive, very, very expensive equipments. But if you can use UV, UV is very cheap. But unfortunately, metals cannot directly absorb UV. And this is the place where we make use of a complexation reaction. We add reagents to this water sample, and these reagents are called element-specific reagent. If I want to detect iron in water, I'll be adding a certain reagent, 1 ton phenanthrolene. I mean, this will react only with iron, form a complex, and this complex has got UV absorption. And from the absorption of the complex, I can calculate very precisely what is the content of iron in water. So for this basically, we have got a separate model of instrument for this, doing this. That model is called 1280, which we are promoting in those type of areas. Fertilizer companies, huge petroleum and petrochemical companies, and power generation companies. We are not promoting it anywhere. I mean, so basically for design for water analysis. And the last 2-3 slides, UV instrumentation. I told you about single beam and double beam. Nowadays, you will find only double beam because people all prefer a double beam instrument. So single beam, what you see on the top is a single beam instrument. There's a source and the light is coming to monochromator, and monochromator will select the required wavelength. It will pass to the sample compartment. From the sample compartment, it will go to the detector and then your software and PC. Okay. So here you have to measure the standard first and then sample first. Standard and sample, you have to separately measure. Okay, because there is only one path, there is only one beam, and that is why it's known as single beam. And in double beam, below what I have shown is double beam. So whatever light is coming from the source, it will come to the monochromator. Monochromator will select the required wavelength, and then using a chopper, we will divide this into two parts. We will divide this light, this monochromatic light, into two parts. One part will pass to the reference side and another part will pass to the sample side simultaneously. So you are keeping the reference and sample simultaneously inside the sample compartment. One is a reference side and one is a sample side. And the monochromatic light is passing simultaneously through the reference side and sample side. And once it comes out, once the light comes from the reference side, once it comes from the sample side, there are two. Either you can use two different detectors, one for reference detection and one for sample detection. That possibility is there. Otherwise, these two beams can be again recombined with another chopper, and then it can pass to a single detector. And then it go to your PC and software. This is called a double beam equipment. Nowadays, people all prefer a double beam equipment. What is the reason? Advantages of a double beam system. First of all, let us look at single beam. Single beam advantages, high energy throughput. People say we are not dividing the light, the entire light is passing through the sample. Agree. But problem is unstable baseline. Any difference in the variation in the voltage or incoming electrons can affect your stability of the baseline. And so you get an unstable baseline. It can affect the measurement to a great extent whenever there is a variation in the electrical voltage or inherent electronics, all those. And fewer moving parts, less expensive. A disadvantage is this one. No reference correction. If there is a change in the sample, it should be automatically corrected with the help of a reference. That is not available in the case of a single beam. No reference correction, unstable baseline, major disadvantages. But when you look at the double beam, advantages more sensitive, more stable, and any fluctuation will be automatically corrected because there is a reference inside. The measurement is done simultaneously on reference and sample. So whatever changes are taking place in the sample because of any fluctuation, the same thing will be nullified because of the reference correction. So it gives a very, very stable baseline, and there won't be any fluctuations because of any voltage variations, like etc. And disadvantage is slightly more expensive than the single beam and more electronics and more moving parts. But today, most of the customers prefer a double beam equipment. Okay. And these are some other parts. You can use a D2 for UV. You can use a tungsten filament lamp for visible. Monochromator, there are different types of monochromators. Zerny-Turner monochromator is one of the most popular monochromators which we are using in very high-end. People use in a very high-end system, but in all our systems, we are using a Zerny-Turner monochromator. It's supposed to be one of the most powerful monochromators. And there are different types of detectors. PMT, as I told you, is the most sensitive one, photomultiplier tube. Indium, gallium arsenide, in-gas. We call it as in-gas detector that is used for measurement in NIR region, and then photosilicon diode. So these are the three different detectors. And there are different accessories are available. I am not going to details. Long path length rectangular cell holder is there. As I told you in the beginning, the standard cell is always 1 centimeter by 1 centimeter. But if suppose the customer is having a sample, the concentration is so low, so if you make the measurement in 1 centimeter cell, you will not get the sensitivity. So as per the Lambert's law, if you increase the path length, your absorbance will increase. So we have different path length, long path length cells available, right from 1 centimeter up to 10 centimeter path length cells are available with us. And when you are going for a long path length cell, it should be having the proper holder also. That is called long path length rectangular cell holder. Similarly, constant temperature cell holder. Some of the biotechnological applications, temperature is a very, very important consideration. Temperature should be maintained absolutely. Even 1 degree variation temperature will have drastic effect on the reading. So we have two different types. Either you can go for a locally available thermostatically controlled water bath, which will circulate heated water around the cell inside the instrument. But then you'll have to water circulation and all those hassles will be there. Otherwise, we can offer a TCC 100, which is a Peltier cooling system. No water bath, no water circulation, electronic heating and electronic cooling. Temperature can be maintained from 7 degrees to 60 degrees centigrade constant. Because all the biotechnological applications, the temperature that is required is 60 degrees centigrade. DNA, RNA quantification, protein instrumentations, all those. Then we have got integrating spheres, different types of integrating spheres for solid sample analysis, ISR 2600, ISR 2600 plus, ISR 603, all those. So these are different attachments. And do's and don'ts, basically, I'm not giving them because you all know some of the important things to be, but not this is my last slide, of course. I mean, when you are drying, many times even people commit mistakes, even in industry today. They clean the cuvette and then for quick drying, they keep it in the oven. That's a very wrong practice. You should never dry the cuvette in the oven. Even though glass and quartz are basically non-conducting material, but when you are keeping inside a heated oven, minute variations, expansion, thermal expansion can take place, which will alter the path length. That will affect your quantitative result. So rinse the cuvette, clean it well, rinse the solvent, just keep it on your table for 5 minutes and it will automatically dry. You can give a rinse with acetone or something, it will automatically dry. And if you feel after so many usage, if you feel there is something sticking to the side, maybe on a weekend, you can put this cuvette in a dilute chromic acid solution overnight. Chromic acids are very, very powerful oxidation systems. And the next day morning when you come, you can take it out, wash in water, plenty of water, finally rinse it with distilled water, and give a final rinse with good quality acetone, air dry, keep it on the table. And this will become very, very, very clean. And when you are taking the solution also inside the cuvette, don't fill the cuvette to the totally. It will start overflowing. And overflowing, then people start, I have seen people cleaning the sides of the cuvette with a lab apron, lab coat. And lab apron is made of cotton. And when you are rubbing the sides, the polished sides of a cuvette with a lab coat, you are actually creating small, minute scratches on the surface. And these type of scratches are coming, it becomes a point of your light will get diverted. I mean, so the amount of light that is going to the detector will reduce. That means the sensitivity will. So never do that. We tell the students, we tell the people in the industry, never clean the sides of a cuvette with your lab coat. Take a lint-free tissue paper which is very, very smooth and just rinse once, that's all. You need to fill the cuvette only up to about 75% of the total volume. The total cuvette volume is around 3 ml. You need to fill it up to around 75%, not more than that. So the overflowing will not take place. And these are some of the and when you are starting the instrument, there is an initialization program. That means an initial check automatically. The instrument will check whether all the hardware, everything is working okay. And when you are doing that, when you are starting the instrument, during this auto check, you should never open the instrument cover. And second thing, when you are that's called the initialization step. So when you are starting the instrument in the morning, see that nothing is there inside the sample compartment. If something is there, please remove that. And then you go for an auto check. Okay, so these are the things which we have to, people have to follow. And finally, these are the different models from Shimadzu. These are the UV visible models. I told you UV 1280 is purely for water analysis. We are not promoting it anywhere else. It is a 5 nanometer single beam equipment. Only 1280 is a single beam equipment. Rest all equipments are double beam equipments. UV 1780, UV 1900 is the most prominent pharmaceutical model. It is conforming to all the pharmaceutical requirement. validation requirements, everything. 1900 we are selling like hotcake in pharmaceutical industry. 1900I. Then we have got two high-end models, 2600 and 2700. 2700 is also a research model. It contains two monochromators inside. All other instruments contain only one monochromator, but 2700 contains two monochromator because we want to reduce the sterilite. In 2700, the sterilite specification is 0.00001%. Why? We are able to achieve that level because of two monochromators. But the instrument becomes very expensive because of, and it's only promoted for research activities. Routine applications in pharmaceutical industry, 1900 is an undisputed model, 1900I. Then we have got two models, which is UV visible NIR model. One is 3600I and one is 3700I. 3700I is a floor model. You have to keep it on the floor. Extremely costly equipment, because this can go to measurements up to in the UV, the vacuum UV range also. I told you, normally we cannot go below 190 nanometer because most of the equipments are starting from 190 nanometer, but this particular equipment can go much below that. And that area is known as vacuum UV region. So UV 3700 is very, very expensive model, purely a research model. It can even work in the vacuum UV region. So these are the two important UV visible NIR models from Shimadzu. And this is my contact details, my email ID, and my mobile number. So any other participant in future wants to contact me, either my mail or on my mobile, you are always welcome. Whatever I know, I can share with you. Otherwise, I can get the information from Shimadzu. I mean, last time also I told you, any of you wants to visit our customer service lab in Mumbai, you are most welcome. You can contact us and we'll be too happy to arrange that. Okay. So with this, I finish my small discussion on UV. Thank you very much. I thank all the participants for the patient hearing, and because it's already 1:30, it's your lunch time. So thank you once again, and wish you all the best. Over to you, sir.
Yes, thank you, sir. Madam.
Yes. Thank you very much, sir, for your informative session. Now the session is open for discussion. So if any questions are there from our participants, they are most welcome. Any questions? Okay. I think there are no questions from our participants. So thank you very much, sir. Girijan, sir.
My pleasure, madam. My pleasure.
For your excellent presentation on basics and applications of UV visible spectroscopy. The session was really very informative. You have explained the basic concepts of UV visible spectroscopy in detailed manner. Also, you have enlightened the applications of UV spectroscopy in agricultural, biological, industrial, as well as environmental fields. Also, the limitations of UV spectroscopy you have covered. Then you have also elaborated the instrumentation part of this technique very nicely and in simplified manner. You also pinpointed the do's and don'ts in UV operation, which will be quite helpful for us in our future studies. So thank you very much, sir, for sparing time and sharing your knowledge and expertise in analytical field with us. Thank you, sir. Now I request Professor Gonzari, sir, to present the certificate of appreciation to Girijan, sir. Gonzari, sir. So thank you very much, sir.
Thank you, madam. Thank you, sir. I accept the certificate gracefully, and it was my pleasure to be a part of this program. At least I was lucky enough to be with all the participants on 2 different occasions. Yes. And for 2 different topics. I hope I was able to give some information on 2 different topics. I hope to meet you all again sometime.
Yes. Sure, sir.
Yes,
Very
sir. Thank you very much.
Thank you, sir. Thank you.
Thank you.
See you soon. Thank you.
So with this, we conclude today's second session. The next session will start sharply at 2:30 p.m.